Badan Litbangkes memberikan dukungan dalam penanganan pandemi COVID-19 melalui jejaring laboratorium pemeriksaan sejak tanggal 16 Maret 2020 dengan kegiatannya penguatan kapasitas laboratorium di daerah-daerah dalam melakukan pemeriksaan spesimen COVID-19. Badan Litbangkes juga melakukan surveillance whole genome sequencing yang menghasilkan pemetaan varian virus SARS-CoV-2 yang terdeteksi di Indonesia. Riset monitoring dan evaluasi vaksin COVID-19 yang dilakukan dalam 7 topik. Manajemen logistik dilakukan untuk mengetahui perencanaan, pelaksanaan, serta sistem pencatatan dan pelaporan vaksinasi dari tingkat pusat, provinsi, kabupaten kota, hingga puskesmas. Efektivitas vaksin. Kegiatan ini dilakukan terhadap 71.455 tenaga kesehatan di DKI Jakarta. Survei attitudinal. Survei ini dilakukan untuk mengetahui penerimaan masyarakat terhadap vaksinasi COVID-19 di Indonesia. Riset durasi proteksi. Riset ini dilakukan untuk mengetahui kadar antibodi sebelum dan setelah vaksinasi, faktor yang mempengaruhi terbentuknya antibodi, dan mengetahui kejadian COVID-19 pada masyarakat yang mendapatkan vaksin Sinovac atau AstraZeneca di lokasi studi yang telah ditetapkan. Riset efek vaksinasi terhadap transmisi COVID-19. Riset ini dilakukan untuk mengetahui seberapa besar kemampuan vaksin COVID-19 dalam menurunkan transmisi. Seroprevalence Survey dilakukan untuk mengetahui jumlah individu dalam suatu populasi yang menunjukkan hasil positif COVID-19 berdasarkan spesimen serologi. Riset manajemen limbah vaksin dilakukan untuk memperoleh data dan informasi tentang hasil pengelolaan limbah vaksinasi COVID-19 dan variasi vaksin wastage rate di berbagai wilayah. Badan Litbangkes juga melakukan pengembangan riset vaksin merah putih. Tujuannya adalah dalam rangka pemenuhan kebutuhan dalam negeri sebagai upaya peningkatan ekonomi negara sesuai arahan Bapak Presiden Indonesia. Bekerja sama dengan perguruan tinggi, lembaga riset, dan sektor swasta untuk mengembangkan produk vaksin COVID-19.
Badan Litbang Kesehatan sebagai bagian dari Kementerian Kesehatan Republik Indonesia bertugas melakukan penelitian dan pengembangan di bidang kesehatan. Dengan kekuatan satuan kerja pusat dan daerah serta jejaring penelitian nasional, Badan Litbang Kesehatan berusaha membantu mengatasi berbagai masalah kesehatan melalui berbagai macam penelitian, baik penelitian skala nasional, riset khusus, riset dasar, riset operasional, maupun intervensi sebagai dasar kebijakan pembangunan kesehatan.
Kami cukup senang, cukup bangga ya dengan bekerja sama dengan Litbangkes, di mana banyak produk-produk hasil penelitian ini yang kita bisa pakai dalam pengembangan kesehatan di Indonesia.
Badan Litbang Kesehatan selama ini telah memberikan layanan publik berupa layanan data, Material Transfer Agreement, review proposal dalam hal etik, hak kekayaan intelektual, pemeriksaan spesimen, kultur, herbarium, simplicia, dan jamu tersaintifikasi. Juga wisata ilmiah, penelusuran pustaka, penerbitan buku, jurnal ilmiah, layanan arsip, dan rekomendasi kebijakan. Dengan dukungan 16 satuan kerja yang tersebar dari Aceh sampai Papua, kami berusaha membantu penanggulangan pandemi COVID-19 dengan berbagai kegiatan, di antaranya penelitian dan kajian, penerimaan dan pemeriksaan sampel, koordinator dan pembina jejaring laboratorium pemeriksaan sampel COVID-19, dan distribusi logistik COVID-19. Kegiatan tersebut perlu dukungan penuh dari layanan publik Badan Litbang Kesehatan, terutama dalam menanggapi, menyampaikan informasi, dan memberikan layanan kepada masyarakat. Laboratorium Badan Litbang Kesehatan merupakan laboratorium rujukan dalam layanan pemeriksaan sampel COVID-19. Hasil pemeriksaan laboratorium yang dilakukan jejaring lab dimasukkan ke dalam aplikasi All New Record, sebuah layanan satu pintu dalam pelaporan lab COVID-19 yang dikoordinir oleh Badan Litbang Kesehatan.
Yang dimaksud dengan All Record PC19 adalah aplikasi yang berisi tentang identitas dari orang yang diperiksa di laboratorium tersebut, kemudian hasilnya. Nah, hasilnya ini sangat penting karena inilah yang akan menjadi dasar untuk tata kelola dalam pelayanan kesehatan atau klinis maupun untuk melakukan penelusuran ataupun tracing apabila ditemukan kasus.
Layanan penelusuran informasi terkait COVID-19 disediakan secara online dan selalu kami update. Layanan etik pun dibanjiri dengan berbagai permintaan untuk penelitian terkait COVID-19. Kami juga berperan serta melalui webinar untuk berbagi informasi terbaru terkait COVID-19, baik skala nasional maupun internasional. Melalui media sosial, Badan Litbang Kesehatan turut andil dalam menyampaikan informasi serta menangkal berita hoax terkait COVID-19.
Pelayanan publik Badan Litbang Kesehatan dapat diakses secara online atau datang langsung ya ke gedung pelayanan publik Badan Litbang Kesehatan.
Untuk keterangan lebih lanjut, layanan publik Badan Litbang Kesehatan dapat diakses melalui alamat-alamat website berikut ini.
Good morning, Your Excellency Minister of Health, Republic of Indonesia, Vice Ministry of Health, Honorable officials of the Ministry of Health, ministries and institutions in Republic of Indonesia, distinguished speakers, moderators, respected audiences, ladies and gentlemen. My name is Uli, your Master of Ceremony for today. In this beautiful morning, we welcome you all to attend this webinar hosted by the National Institute of Health Research and Development, Ministry of Health, Republic of Indonesia. Before we proceed further this morning, I would like to invite ladies and gentlemen to properly pose in the upright positions for the singing of our national anthem, Indonesia Raya. Thank you, everyone. All of you may take your comfortable positions. Without further ado, I would like to invite the Acting Director General of National Institute of Health Research and Development, Dr. Nana Mulyana, to deliver the committee report. Bapak Nana, jika Bapak sudah siap untuk memberikan laporan, kami persilahkan untuk memulai Bapak.
Ya, baik. Yang terhormat Bapak Menteri Kesehatan Republik Indonesia, Bapak Budi Gunadi Sadikin, Bapak Wakil Menteri Kesehatan, Bapak Dr. Dante, Saksono Harbuwono, Bapak Ibu Eselon 1, Eselon 2 di lingkungan Kementerian Kesehatan, pembicara dari Indonesia, Pak Prof. Tjandra Yoga, Dr. Ristiyanto, pembicara dari beberapa negara, Dr. Raymond, Dr. Adam P. Robert, Dr. Ting. Thank you for being willing to be a speaker today. Moderator, Dr. Yanri dan Ibu Dr. Rita, Bapak Ibu sekalian yang berbahagia. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, good morning ladies and gentlemen. Salam sejahtera untuk kita semua. Mari kita panjatkan puji dan syukur kehadirat Allah Subhanahu wa Ta'ala, Tuhan Yang Maha Esa, yang telah memberikan kesehatan dan kekuatan. Pada hari ini kita bisa ikut bersama-sama dalam acara webinar Webinar hari ini diselenggarakan dalam rangka Hari Kesehatan Nasional ke-57 tahun 2021 dan topik webinar hari ini adalah surveillance berbasis laboratorium dalam mendukung sistem ketahanan kesehatan nasional. Tema ini sejalan dengan transformasi kesehatan yang saat ini sedang berlangsung di Kementerian Kesehatan yang akan memperkuat surveillance berbasis laboratorium yang sangat dibutuhkan untuk mengatasi pandemi COVID-19 maupun untuk mengantisipasi kejadian new emerging disease di masa akan datang sehingga akan memperkokoh kita dalam menyiapkan sistem ketahanan kesehatan nasional kita. Bapak Ibu sekalian yang saya hormati, pada webinar hari ini akan disampaikan materi-materi oleh para ahli di bidangnya. Dalam hal ini adalah yang terkait dengan genome sequencing baik dari Singapura, Inggris, Belanda, dan dari Indonesia. Pembicara utama adalah Bapak Wakil Menteri Kesehatan. Nanti akan bicara Dr. Raymond Lin from Singapore dan Dr. Adam V. Robert dari Inggris yang akan menyampaikan surveillance dan deteksi antimikrobial resistance. Dr. Dirk Eggink dari Belanda akan menyampaikan bagaimana surveillance SARS-CoV-2 di Belanda, dan pembicara dari Indonesia Prof. Tjandra Aditama akan menyampaikan terkait surveillance TB, dan Dr. Ristiyanto akan menyampaikan bagaimana surveillance leptospirosis. Webinar pada hari ini akan dipandu oleh moderator kita Ibu Dr. Yanri Wijayanti Subroto dan Ibu Dr. Rita Kusriastuti. Bapak Ibu sekalian, kami menyampaikan terima kasih dan penghargaan yang setinggi-tingginya pada semua pihak yang membantu terselenggaranya kegiatan webinar hari ini. Selanjutnya mohon perkenan Bapak Menteri Kesehatan untuk membuka acara webinar ini secara resmi. Welcome to the webinar. My God bless us all. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Nana Mulyana. Next, I would like to invite Your Excellency, Vice Minister of Health, Dr. Dante Saksono Harbuono, to deliver the keynote speech. Your Excellency, Dr. Dante, are you ready?
Yes, I'm ready.
Okay.
Thank
you,
you.
Doctor.
Assalamualaikum warahmatullahi wabarakatuh. Bisa dimulai?
Yes, we can hear you. The floor
Silakan,
is
Bapak.
yours.
Ya. Assalamualaikum warahmatullahi wabarakatuh, Bapak dan Ibu sekalian yang saya hormati. Saya ucapkan terima kasih atas penyelenggaraan simposium ini, dan saya mengucapkan terima kasih atas diberikan kesempatan kepada saya untuk menyampaikan beberapa hal penting tentang bagaimana kita melakukan ketahanan dan strengthening untuk memberikan beberapa hal yang mungkin akan bermanfaat pada perjalanan simposium ini. Izin saya untuk slide show. Ya, the strengthening laboratory-based surveillance dari National Health and Research Development Vice Minister of Health Republik Indonesia. Bapak dan Ibu sekalian, saya akan berbicara mengenai 3 hal utama, yaitu situasi pandemi yang kita hadapi sekarang, kemudian deteksi SARS-CoV-2 varian, dan strategi untuk meningkatkan strengthening laboratorium berbasis surveillance. Ini ada strategi yang kita tetapkan untuk menangani pasien COVID-19, dan deteksi tersebut merupakan 3 strategi utama, yaitu deteksi, terapeutik, dan vaksinasi. Hal yang paling penting dalam deteksi adalah meningkatkan secara epidemiologis tes dibandingkan dengan screening tes dan peningkatan kontak tracing pada para pasien, pada para keluarga dan pada para kontak erat pasien-pasien yang terkonfirmasi COVID-19. Kemudian juga yang penting adalah memberikan genomic surveillance karena partikular area yang memperlihatkan bahwa peningkatan kasus signifikan secara confirm akan terdeteksi dengan melakukan surveillance pada kelompok yang perlu dilakukan screening. Dan screening tersebut menjadi sangat penting dalam bagian keseluruhan tata laksana secara epidemiologis, secara klinis bagi pasien-pasien di tingkat populasi. Selain Bapak-bapak, ibu-ibu sekalian, dengan melakukan kegiatan deteksi yang baik, maka kita bisa melihat bahwa peningkatan kasusnya yang tadi yang meningkat dengan tajam pada kira-kira bulan Juni sampai 4,5 kali lipat, sekarang sudah mulai menurun. Hingga tadinya ketika puncak tersebut kita punya 56.757 kasus, saat ini hanya mencapai 339 kasus baru per hari di Indonesia. Slide. Bapak Ibu dan sekalian, bagaimana deteksi SARS-CoV-2 varian ini menjadi sangat penting bagi deteksi pasien-pasien COVID-19 di populasi. Ini adalah peningkatan kasus-kasus baru di negara Eropa, particularly pada beberapa yang sudah mempunyai higher rate of vaccination, tetapi juga masih terkendala dengan terjadinya lonjakan kasus. Misalnya di Amerika Serikat, bahwa deteksi secara surveillance dan genomik menunjukkan bahwa peningkatan case pertama disebabkan oleh varian Alpha. Kemudian pada wave kedua yang dominan ada varian Delta. Jadi kelihatannya di dalam populasi peningkatan dan peningkatan kasus itu terutama dikontribusikan oleh varian Delta. Kemudian di Israel, di mana tempat yang sudah memberikan vaksinasi hampir 97% dari populasi. Tetap saja begitu ada varian Delta yang masuk ke dalam populasi tersebut, maka peningkatan kasus juga terjadi. Demikian juga di Inggris. Ketika di Inggris itu sudah masuk wave pertama disebabkan oleh varian Alpha, tetapi kemudian ada varian Delta, maka varian Deltanya tersebut yang akan menyebabkan dominasi kasus, sehingga peningkatan kasus itu terjadi di Eropa akibat varian-varian Delta ini. Dan coba kita lihat bagaimana data secara spesifik dari UK bahwa di dalam varian Delta terdapat subvarian yang menyebabkan menjadi dominan. Antara lain sekarang yang menjadi dominan adalah AY4.2, dan AY4.2 disinyalir dan diduga menjadikan salah satu penyebab kenapa varian dan angka peningkatan kasus di Inggris secara epidemiologis itu meningkat. Ini disebabkan karena varian tersebut lebih banyak terdeteksi di Inggris dibandingkan dengan varian Delta origin yang ada di sebelumnya. Slide. Bapak dan Ibu sekalian, pada saat kita melakukan surveillance system dengan laboratorium dan genomic sequencing test, maka kita lihat bahwa yang tadinya kita bulan Januari hanya bisa memeriksa 338 tes untuk genome sequencing, saat ini kita bisa mencapai 8.415 genome sequencing sampai tanggal 21 Oktober. Jadi proporsi delta varian pun juga kita evaluasi, dan Bapak Ibu bisa lihat bahwa proporsi delta varian yang ada di Indonesia tersebut meningkat, dan peningkatan kasusnya disebabkan karena varian Delta ini menjadi dominan. Dan kita menyadari bahwa spreading capacity of varian Delta ini menjadi salah satu faktor penentu dalam deteksi new variant concern yang berpengaruh pada kejadian kasus yang meningkat di berbagai tempat. Slide. Dan kita lihat bahwa laboratorium surveillance menjadi sangat penting untuk memonitor COVID varian dan melakukan sirkulasi across the country. Dan kita lihat bahwa ada 2 varian yang saat ini selain varian Delta yang menjadi varian of interest, antara lain ada varian Lambda dan varian Mu. Dan varian Lambda ini dikategorikan sebagai varian of interest, begitu juga oleh varian Mu. Ini pertama kali dideteksi di Peru tahun 2020 Desember, sedangkan varian Mu terdeteksi di Kolombia tahun 2021 bulan Januari. Tapi di negara kita kedua varian tersebut belum ditemukan, sehingga dari 41 negara yang terkonfirmasi pasien Lambda dan 53 kasus yang terinfeksi dengan varian Mu di Indonesia, yang dominan pada saat terjadinya eskalasi kasus beberapa waktu yang lalu adalah varian Delta yang lebih infeksius daripada varian Lambda dan varian Mu. Karena itu mudah-mudahan tidak terjadi lagi mutasi yang kita deteksi pada genomic surveillance laboratorium yang menyebabkan peningkatan kasus pada pasien-pasien di populasi yang semakin meningkat untuk beberapa negara yang masuki wave-wave berikutnya. Slide. Bapak dan Ibu sekalian, lebih lanjut bahwa laboratorium surveillance system juga harus dibangun sebagai tools untuk meningkatkan emerging dan emerging infection disease, tidak saja pada kasus-kasus COVID, tetapi juga pada beberapa kasus. Kita harus meningkatkan ketahanan kita untuk melakukan permodelan laboratorium yang bisa mendeteksi varian tersebut merupakan varian yang harus kita perbaiki. Bapak dan Ibu sekalian, slide berikutnya, bagaimana strategi kita untuk meningkatkan kapasitas surveillance dari kasus-kasus tersebut di dalam kerangka pembentukan dan peningkatan kapasitas laboratorium surveillance. Slide. Dan kita lihat bahwa kita mengidentifikasi berbagai isu yang berkaitan dengan related based surveillance. Yang pertama adalah bahwa low score health emergency preparedness dan respon di Global Health Security Index menunjukkan bahwa real time surveillance capacity baru mencapai ranking 90 untuk Indonesia, dan emergency preparedness response planning baru mencapai ranking ke-67. Ini kenapa? Ini disebabkan karena sistem organisasinya yang terlalu rumit. Ada virus influenza yang dilakukan di laboratorium tertentu, TB di laboratorium tertentu, dan polio di laboratorium tertentu. Begitu juga dengan pemeriksaan-pemeriksaan lainnya. Secara ideal di Indonesia, laboratorium network itu harus tidak terfragmentasi, sehingga primary healthcare, prevention, regional, nasional, dan ideal itu mengerucut bentuknya seperti segitiga piramid ini, sehingga semua laboratorium bisa mengerjakan deteksi pada kasus-kasus yang spesifik tersebut, dan akhirnya membuat kita bisa melakukan data terintegrasi yang lebih baik. Bapak dan Ibu sekalian, delay surveillance summary dari apa yang kita lihat dari kasus-kasus ini adalah bahwa yang paling penting adalah daily system surveillance report system ini mesti diperbaiki. Laboratorium based surveillance system masih bervariasi sehingga tidak terintegrasi. Ini harus dibuat lebih terintegrasi lagi. Dan formal system emergency health workforce voluntary juga harus diperkuat, sehingga kita memiliki kapasitas laboratorium yang lebih tidak terfragmentasi di seluruh Indonesia. Ini PR ke depan bagi Kementerian Kesehatan dari Litbangkes, sehingga model-model yang ideal dengan piramid network Indonesia yang tadinya terfragmentated menjadi terselesaikan. Slide. Dan kita tahu bahwa dengan membangun sistem tersebut, maka healthcare system transformasi bisa terjamin, dan salah satunya adalah strategi emergency response resiliency, di mana National Network Laboratorium Surveillance, emergency response tabletop to every time for preparedness lebih terjamin, sehingga kita bisa melakukan beberapa hal yang bersinggungan dengan transformasi kesehatan. Memperkuat laboratorium merupakan bagian dari peningkatan dan upaya untuk memberikan outcome transformasi sistem kesehatan yang baik di negara kita. Slide. Dan strategi kunci dari peningkatan laboratorium surveillance di Indonesia ini adalah melakukan double PCR yang tadinya dari 446 laboratorium menjadi 921. Laboratorium surveillance harus bisa mengevaluasi dan melakukan reassessment surveillance laboratorium capacity. Laboratorium kesehatan referral system design harus dibentuk, laboratorium quality standarisasi dan human resource capacity harus dibentuk, dan implementasi digital harus merupakan salah satu reporting yang terintegrasi sehingga bisa dilihat secara real time dari daerah sampai pusat. Untuk menentukan langkah-langkah strategis untuk membangun penguatan sistem laboratorium dan membantu bagaimana mencegah terjadinya peningkatan kasus-kasus berikutnya. Slide. Sebagai konklusi saya bisa sampaikan bahwa walaupun kita sekarang berada dalam situasi yang menurun kasusnya, tapi kita harus mempersiapkan diri untuk menjadi kemungkinan terjadinya another wave of COVID-19. Mudah-mudahan COVID-19 tidak masuk ke wave ketiga di Indonesia karena kita sudah melakukan persiapan dan baik di tingkat publik, kebijakan, laboratorium, terapi, perawatan, dan sebagainya, sehingga wave tersebut tidak terjadi. Tentu ini butuh peran dari masyarakat selain teman-teman dari laboratorium. Deteksi merupakan bagian strategi yang terpenting juga, menemukan new case, new variant circulated di seluruh dunia menjadi salah satu tumpuan dan harapan kita kepada laboratorium-laboratorium yang ada di Indonesia. Dan laboratorium surveillance system tidak saja memberikan effort bagi konten current pandemic, tapi juga memberikan respon untuk emerging dan re-emerging infection disease, sehingga pasien-pasien, kondisi populasi, kondisi epidemiologis bisa terkontrol dengan ditemukan varian baru. Kementerian Kesehatan telah melakukan map out bagi plan reform untuk kesehatan, termasuk diantaranya adalah penguatan laboratorium based surveillance. Begitu yang bisa saya sampaikan, mudah-mudahan bisa menjadi bahan diskusi yang baik untuk pertemuan hari ini. Semoga kesempatan dan pertemuan pada hari ini bisa terlaksana dengan baik. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh. Om Santi Santi Om.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih Dr. Dante atas keynote speech yang tadi sudah disampaikan. Now we officially proceed to the core sessions of this webinar, but before we start, we would like to show the tapping video from His Excellency Ministry of Health of Budi Gunadi Sadikin. But before we show the tapping video, we would like to invite all the panelists to take shot photo together. Bapak Ibu, sebelum kita mendengarkan tapping video dari Bapak Menteri Kesehatan, kami ingin mengundang Bapak Ibu para panelis untuk berfoto bersama. Ya, mohon kepada Bapak Ibu para narasumber panelis untuk mengaktifkan videonya. To all speakers, we would like to invite you to take shot together. So please kindly open your video. Okay, in 1, 2, 3, we would like to take photo together. Host, please help. 1, 2, 3. Okay. Bapak Ibu para undangan. Okay, yeah. Okay. Again. 1, 2, 3. Okay, thank you Bapak Ibu semuanya. Selanjutnya kita akan memutar video tapping dari Bapak Menteri Kesehatan. Kepada host dipersilahkan untuk memulai.
Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semua. Om Swastiastu, Namo Buddhaya, salam kebajikan. COVID-19 semakin menyadarkan kita terhadap pentingnya salah satu fungsi kesehatan masyarakat untuk menghadapi ancaman epidemi dan pandemi penyakit, yaitu kewaspadaan dini dan respons yang cepat atas ancaman krisis kesehatan. Untuk itu, Kementerian Kesehatan terus berproses untuk melaksanakan transformasi sistem kesehatan, khususnya transformasi sistem ketahanan kesehatan. Selain kemandirian obat dan alat kesehatan, juga termasuk jejaring surveillance, khususnya surveillance berbasis laboratorium. Peningkatan kapasitas laboratorium, baik infrastruktur, tata kelola, sistem informasi, jejaring, dan sumber daya manusianya adalah hal yang harus terus menjadi perhatian kita bersama. Surveillance berbasis laboratorium ini termasuk dalam kapasitas inti International Health Regulation yang harus dimiliki oleh semua negara, termasuk Indonesia. Hal ini tentunya sangat berperan dalam mendeteksi penyebab kejadian kedaruratan. Saya mengapresiasi kinerja Badan Litbangkes sebagai bagian dari pemerintah pusat yang terus berperan bersama kita semua dalam menangani pandemi COVID-19 dengan melaksanakan program-program prioritas dan inovatif sejak awal pandemi. Dukungan jejaring laboratorium pemeriksaan spesimen dan kegiatan surveillance whole genome sequencing COVID-19 yang dikoordinasikan dan baik dari pusat hingga daerah ini merupakan bentuk kewaspadaan dan deteksi dini COVID-19, serta menjadi cikal bakal jejaring kewaspadaan dan deteksi dini untuk patogen penyebab pandemi-pandemi berikutnya, yang tentunya kita harapkan tidak akan terjadi, namun kita harus tetap siap dan waspada. Selamat mengikuti webinar. Semoga kita menghasilkan hal-hal bermanfaat untuk bangsa kita di tengah perjuangan kita berdamai dengan pandemi. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh. Om Santi 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 Om.
Respected audiences, ladies and gentlemen, by the end of the taping video from His Excellency Ministry of Health, we cordially open this webinar. There are some informations that I would like to share with you that for attendance in the Zoom webinar, we have provided a translator to translate from Bahasa Indonesia to English or backwards. And you can find the feature in your Zoom screen. Zoom attendants can click the interpretation facility on your screen to listen to the original language delivered by the speakers. And then by the end of this webinar, there is an evaluation form will be given 15 minutes before the end of this webinar. Patiently stay tuned for it, and we will use the information that you filled in the form for your e-certificate purposes. This webinar will be divided into 2 sessions. The first 3 speakers will be moderated by Dr. Yanri Wijayanti Subroto. And Dr. Yanri is the lecturer in the Faculty of Medicine, Public Health and Nursing Gadjah Mada University. And she is an internist in Dr. Sarjito Hospital, Yogyakarta. She has published a lot of scientific publications in infectious diseases in reputable journals such as Annals of Medicine and Surgery, AIDS Education and Prevention, and Clinical Pharmacokinetics. Ladies and gentlemen, allow me to invite Dr. Yanri and pass these first sessions to her. Dr. Yanri? Yeah, are you ready?
Yes.
Good morning, Dr. Yanri. Yes.
Good morning.
Okay. Okay, you can have the floor now.
Okay, thank you, Mauli. My voice kind of dropped, I think. Okay. Selamat pagi semuanya, salam sejahtera dan salam sehat untuk kita semua. Semoga semuanya bahagia. Good morning to all the speakers that comes from Singapore, the Netherlands, and England. Even though we don't know where you are at the moment, whether you are staying in your country or you are somewhere else. So thanks to the advance of technology that we can meet from anywhere around the world. So it is an honor for me to host this session. Thank you to M.Kes for asking me to join this important webinar. I think this is a very important webinar. So the first speaker will be Dr. Raymond Lin, who is the director at the National Public Health Laboratory, National Center for Infectious Diseases in Singapore. Good morning, Dr. Lin. Hello, Dr. Lin. Are you already in front of the camera? Okay. After Dr. Lin, we will hear from Dr. Dirk Eggink. I hope I pronounce it right. He is the Senior Scientist Virology, Center for Infectious Disease Control, National Institute for Public Health and the Environment. It is called RIVM in the Netherlands because I used to study my PhD in the Netherlands. So I kind of know the RIVM and I think I have visited there. So he is in RIVM in the Ministry of Health, Welfare and Sport in the Netherlands. And he is also Assistant Professor Laboratory of Applied Evolutionary Biology, Department of Medical Microbiology. And then the last one from our session this morning will be Dr. Adam P. Roberts, PhD, and current employment is in the Liverpool School of Tropical Medicine. So without further ado, I would like to invite Dr. Lin, already in the room. His education is the Royal College of Pathologists of Australasia. And then selected scientific publications is the SARS-CoV-2 infections among travelers returning from Wuhan, China, which was published in 2020. And then the second one is the epidemiologic features and clinical course of patients infected with SARS-CoV-2 in Singapore. I think this is very, very important, very recent, and it is very now. Again, I would like to invite Dr. Lin.
Ibu Yanri, izin,
Yes.
I think he mental.
Oh,
Oke, ini
okay.
kami coba sedang menghubungi beliau lagi. Tadi sudah join sebenarnya sebelum kegiatan, namun kami cek lagi di list list panelis sepertinya ini. Kami sedang mencoba menghubungi kembali.
Oke. Siap. Bapak dan Ibu, mungkin saya bisa membacakan CV yang lain, jadi supaya nanti ketika Dr. Lin masuk kita bisa langsung menuju ke Dr. Dirk. Dr. Dirk, I guess we will wait for Dr. Lin to join again, to rejoin the room. But I'll just kind of briefly read your CV. So, if I may know, how do you pronounce your last name? I'm sorry. Dr. Dirk, are you inside the room as well? Terlempar juga kah? Mbak Uli, Dr. Dirk ada di ruangan enggak?
Iya.
Kita lihat saja. Kayaknya kok nggak ada juga ya.
Mohon izin Dr. Yanri, mungkin sambil kita mau mencoba menghubungi lagi para narasumber, kita akan putarkan sebentar video sambil panitia mencoba menghubungi kembali para narasumber.
Iya, sepertinya
Iya,
semua sedang dalam pesawat mungkin ya, Mbak Uli ya, mungkin dalam pesawat menuju ke Jakarta kayaknya.
Iya,
Oke,
sepertinya
kita
begitu.
tunggu.
Oke.
Kita tunggu dengan menonton video.
Ya, kita putarkan video terlebih dahulu. Bapak Ibu mohon berkenan untuk menyimak. Terima kasih.
Dalam situasi pandemi COVID saat ini, kita semua tahu kalau dokter dan perawat adalah tenaga kesehatan yang bekerja keras dan dekat dengan resiko. Tapi belum banyak yang tahu kalau ada pejuang lain yang bekerja dalam senyap. Mereka adalah tenaga laboratorium pemeriksa spesimen di Badan Litbang Kesehatan.
Tidak pernah terbayangkan sebelumnya kami akan melewati hari-hari seperti ini. Hari-hari yang penuh dengan sampel spesimen yang terus berdatangan tanpa kita tahu siapa pemiliknya. Sebagai manusia biasa, kami juga merasa sedih. Mungkin masih banyak orang yang belum tahu proses yang sebenarnya terjadi di sini. Hasil tes swab di laboratorium Balitbangkes bisa keluar dalam waktu 2 kali 24 jam. Kami bekerja berpacu dengan waktu, sekalipun itu sampai malam, bahkan harus menginap di laboratorium. Selama spesimen itu ada, kami tak kenal kata libur. Namun, tidak jarang kami tetap dinilai lambat. Kami tetap fokus bekerja sesuai tanggung jawab kami, yaitu mendukung surveillance kesehatan masyarakat. Orang lain boleh tidak tahu siapa kami, tapi kami tahu persis apa yang kami lakukan sehari-hari dalam menjalankan tugas.
Untuk lebaran kali ini memang menjadi lebaran yang berbeda dengan lebaran yang selama ini kita lalui. Untuk kami di sini yang di laboratorium pemeriksaan Badan Litbangkes memang kita tidak menerapkan libur sehari pun.
Kami tidak minta dikenal. Kami hanya ingin dipahami.
Badan Litbangkes memberikan dukungan dalam penanganan pandemi COVID-19 melalui jejaring laboratorium pemeriksaan sejak tanggal 16 Maret 2020 dengan kegiatannya penguatan kapasitas laboratorium di daerah-daerah dalam melakukan pemeriksaan spesimen COVID-19. Badan Litbangkes juga melakukan surveillance whole genome sequencing yang menghasilkan pemetaan varian virus SARS-CoV-2 yang terdeteksi di Indonesia. Riset monitoring dan evaluasi vaksin COVID-19 yang dilakukan dalam 7 topik. Manajemen logistik dilakukan untuk mengetahui perencanaan, pelaksanaan, serta sistem pencatatan dan pelaporan vaksinasi dari tingkat pusat, provinsi, kabupaten kota, hingga puskesmas. Efektivitas vaksin. Kegiatan ini dilakukan terhadap 71.455 tenaga kesehatan di DKI Jakarta. Survei attitudinal. Survei ini dilakukan untuk mengetahui penerimaan masyarakat terhadap vaksinasi COVID-19 di Indonesia. Riset durasi proteksi. Riset ini dilakukan untuk mengetahui kadar antibodi sebelum dan setelah vaksinasi, faktor yang mempengaruhi terbentuknya antibodi, dan mengetahui kejadian COVID-19 pada masyarakat yang mendapatkan vaksin Sinovac atau AstraZeneca di lokasi studi yang telah ditetapkan. Riset efek vaksinasi terhadap transmisi COVID-19. Riset ini dilakukan untuk mengetahui seberapa besar kemampuan vaksin COVID-19 dalam menurunkan transmisi. Seroprevalence Survey dilakukan untuk mengetahui jumlah individu dalam suatu populasi yang menunjukkan hasil positif COVID-19 berdasarkan spesimen serologi. Riset manajemen limbah vaksin dilakukan untuk memperoleh data dan informasi tentang hasil pengelolaan limbah vaksinasi COVID-19 dan variasi vaksin wastage rate di berbagai wilayah. Badan Litbangkes juga melakukan pengembangan riset vaksin merah putih. Tujuannya adalah dalam rangka pemenuhan kebutuhan dalam negeri sebagai upaya peningkatan ekonomi negara sesuai arahan Bapak Presiden Indonesia. Bekerja sama dengan perguruan tinggi, lembaga riset, dan sektor swasta untuk mengembangkan produk vaksin COVID-19.
Badan Litbang Kesehatan sebagai bagian dari Kementerian Kesehatan Republik Indonesia bertugas melakukan penelitian dan pengembangan di bidang kesehatan. Dengan kekuatan satuan kerja pusat dan daerah serta jejaring penelitian nasional, Badan Litbang Kesehatan berusaha membantu mengatasi berbagai masalah kesehatan melalui berbagai macam penelitian, baik penelitian skala nasional, riset khusus, riset dasar, riset operasional, maupun intervensi sebagai dasar kebijakan pembangunan kesehatan.
Kami cukup senang, cukup bangga ya dengan bekerja sama dengan Balitbangkes, di mana banyak produk-produk hasil penelitian ini yang kita bisa pakai dalam pengembangan kesehatan di Indonesia.
Badan Litbang Kesehatan selama ini telah memberikan layanan publik berupa layanan data, Material Transfer Agreement, review proposal dalam hal etik, hak kekayaan intelektual, pemeriksaan spesimen, kultur, herbarium, simplicia, dan jamu tersaintifikasi. Juga wisata ilmiah, penelusuran pustaka, penerbitan buku, jurnal ilmiah, layanan arsip, dan rekomendasi kebijakan. Dengan dukungan 16 satuan kerja yang tersebar dari Aceh sampai Papua, kami berusaha membantu penanggulangan pandemi COVID-19 dengan berbagai kegiatan, di antaranya penelitian dan kajian, penerimaan dan pemeriksaan sampel, koordinator dan pembina jejaring laboratorium pemeriksaan sampel COVID-19, dan distribusi logistik COVID-19. Kegiatan tersebut perlu dukungan penuh dari layanan publik Badan Litbang Kesehatan, terutama dalam menanggapi, menyampaikan informasi, dan memberikan layanan kepada masyarakat. Laboratorium Badan Litbang Kesehatan merupakan laboratorium rujukan dalam layanan pemeriksaan sampel COVID-19. Hasil pemeriksaan laboratorium yang dilakukan jejaring lab dimasukkan ke dalam aplikasi All New Record, sebuah layanan satu pintu dalam pelaporan lab COVID-19 yang dikoordinir oleh Badan Litbang Kesehatan.
Yang dimaksud dengan All Record TC19 adalah aplikasi yang berisi tentang identitas dari orang yang diperiksa di laboratorium tersebut, kemudian hasilnya. Nah, hasilnya ini sangat penting karena inilah yang akan menjadi dasar untuk tata kelola dalam pelayanan kesehatan atau klinis maupun untuk melakukan penelusuran ataupun tracing apabila ditemukan kasus.
Layanan penelusuran informasi terkait COVID-19 disediakan secara online dan selalu kami update. Layanan etik pun dibanjiri dengan berbagai permintaan untuk penelitian terkait COVID-19. Kami juga berperan serta melalui webinar untuk berbagi informasi terbaru terkait COVID-19 baik skala nasional maupun internasional. Melalui media sosial, Badan Litbang Kesehatan turut andil dalam menyampaikan informasi serta menangkal berita hoax terkait COVID-19.
Pelayanan publik Badan Litbang Kesehatan dapat diakses secara online atau datang langsung ya ke Gedung Pelayanan Publik Badan Litbang Kesehatan.
Untuk keterangan lebih lanjut, layanan publik Badan Litbang Kesehatan dapat diakses melalui alamat-alamat website berikut ini.
Thank you, ladies and gentlemen. You have viewed some videos about the NIHRD, sepak terjangnya Badan Litbangkes dalam penelitian dan pengembangan kesehatan. As we observe in the panelists, Dr. Raymond Lin already joined our webinar. I think all of the international speaker, I mean Dr. Dirk and Adam is from different time difference, so they might have jadi mereka baru bisa bergabung pukul 10 karena di negaranya di Netherlands dan di London mungkin masih subuh begitu. Oke, terima kasih Bapak Ibu yang sudah menyimak tayangan video kami. Selanjutnya kita akan mulai ke sesi pertama dari webinar ini. Kami mohon kesediaan Dr. Yanri untuk memulai lagi sesi pertama ini. To Dr. Yanri, the floor is yours.
Thank you, Mbak Uli. Good morning, Raymond.
Good morning.
Hello, Dr. Lin.
Yeah, hello.
Oh, hi. Okay. Good to have you in the room.
Thank you.
Okay. Earlier, a few minutes ago, I kind of already read your CV, but now that you are already on camera, I'll just take a few seconds again to read your CV. Maybe if it's wrong, then you can correct it. So you are now the director of the National Public Health Laboratory, National Center for Infectious Disease in Singapore. That is correct, huh?
Yes, thank you.
Okay. Yes. And the selected publications that's already mentioned in the CV is the SARS-CoV-2. So I guess without further ado, it would be great to have you talking about country experience of SARS-CoV-2 genome surveillance in Singapore. So the time and floor is yours.
Thank you so much, and I'm very pleased to join you, and thank you so much for the invitation. I will now share. Wait, I will now share my screen. Wait a while.
We have around 390 audience around.
Okay.
So.
Okay. It's not appearing on my thing. Wait just a while. Okay.
We already saw it actually.
Oh, okay, okay, yeah.
Yes.
And I just need to.
Yeah.
I need to go to the presentation.
Yes.
Is that okay? Yep.
Yes. Very, very okay.
Okay. Thank you so much. Yes. So, in order to talk about the genome sequencing for the COVID pandemic, I will need to just roughly go through, because at different stages over the past almost 2 years, we use it a bit differently. So I'll show you the row. Can you see the new slides? Yeah. Yeah.
Yes.
So looking at the reported cases, these are the different phases of the pandemic from the beginning, right? So of course, first we had the cases from Wuhan, which came to Singapore on various flights, and then, well, it seems so many, but now you look at it, it's very low numbers, and then after March, we told Singapore residents and all that, please come back before you're stuck overseas, and then we had many of them coming back from Europe, from US, from UK, and they bring back the virus from there. And then we had from April onwards, we had a very big outbreak in our foreign workers who stay in dormitories, and it spread to almost every dormitory in Singapore, affecting over 50,000 cases. So we call it circuit breaker, but it's basically a kind of lockdown for about 2 months, and it came down, right? And once we opened up, people started to travel to Singapore again, and we had another spate of imported cases with some local transmission. And from then, we had very few local transmissions. So when you talk about surveillance, there's not much to survey because no outbreaks since then. And then in May, we had the 6172, which afterwards was called Delta. Then we had a small increase in cases. You can't see on this chart because it's small numbers, and later on a bigger outbreak in the markets, right? Which we managed to control, but I think the numbers kept increasing, and a decision was made by the leaders to say, let's change our tactic, and since the vaccine uptake was quite good, let's relax a bit on the quarantine and other methods. And so our cases went up very high, which is what you would been reading probably in the news, and I think the emphasis now is to prevent people from getting very sick and dying and occupying our ICU. So that's the emphasis now. So we are basically, although we have some social restrictions, basically every 1 or 2 weeks we open up more and opening up travel, so trying to get life back to normal. But yeah, and cases are stabilizing now. It's no more going up this way. Now it's stabilizing around. Yesterday was about 2,000 cases, right? Yeah. So at different stage we use the genome sequencing differently, right? So just to highlight the early part, right? These are the cases from China, from Wuhan at first. It spread a little bit, and these are these imported cases, right? So these are the people we said, Singapore resident, please come home. Okay, then they came home and brought some viruses. We control them. It's okay, quarantine them a bit. But the local one, these are the foreign workers, right? Foreign dormitories, right? So for foreign dormitories, we, with our lockdown or circuit breaker, managed to reduce it, right? Right, and then after March, April, May, in May it went up because of the new Delta. So when Delta came, actually to me it was, I think we were one of the first places outside the Indian subcontinent to actually realize how it behaved, how easily it spread, right? Right. and as of May, the May Delta came in May, so May we were still having a mixture of imported and local cases. Imported cases, all different kinds, you know, mainly from Europe and USA. You know, B117 is mainly Europe, USA. These are South African strain as well, you know, and only beginning to see Delta. Of course, now later on, Delta replaced everything, right? So local cases, you can see none of the imported genotypes really spread much in the community, hardly any, right? So small numbers. Whereas once Delta entered, it spread and became our local cases, right? And so this was the use of our genome sequencing, where we quickly found out what was spreading in our community and whether from the imported cases, what kind of imported cases, so it's main Delta which spread, right? So that was our first use of systematic surveillance to find out early. And I think the lesson here is that, you know, before May, March, April, May, you know, many of the diagnostic companies will say, oh, I've got PCR to find 117 and 3151. Do you want to try the PCR? Don't have to do the sequencing. Our answer is no, we need to do the sequencing, because if you use the PCR for the known genotype, then you won't know when the new one comes, you know what I mean? And in fact, the first PCR for the different genotypes, for the different variants of concern, VOC, did not include Delta. So if you use the PCR, you will not find Delta when it arrives, right? That's why we believe always to do the sequencing, you know, yeah. And of course, after that, in July, we have more local outbreaks. You know, first we had it spread from the airport cases, where they went home and then went to the shopping centers and different shopping centers. So our first round from Delta, after we had some spread in the hospital, but we slowed that down. But after we controlled the one in the hospital, the cases from the airport cluster went to various places, to the schools, to shopping centers all over Singapore. Singapore very small, so all different shopping centers, and increase in the cases, right? And then after July, we had a second one where then from the, you know, what I call food centers or hawker centers, where most people eat there at, you know, day and at night, it was spread from there. And then later on, we had another round. This time it was not related to the airport cases, where it was linked to the place where we bring in the fish, our fisheries, you know, the market where they sell fish. And from there, the food center, the people, when they buy the fish, they go back to the different areas in Singapore and spread to the markets, right? And one of them, they also spread to our karaoke lounges. You know, these are the night places where people go for beer and karaoke and other activities, right? And then so this look very small when compared to today's cases. So a decision was made end of August to say, let's open up more. We don't need to quarantine everybody, and maybe when people are sick, they can. If people are positive, only the sick ones stay in hospital. Maybe those who are relatively well, not so sick, they can go and stay at home and to recover, right? And then so as a result, the emphasis changed on looking after cases in the ICU. So our cases went up since then. Now it's come down to around here, right? Yeah. So okay, I'll tell you the different use of WGS, right? So of course the first one, when the first case came, like most new diseases, our first case, it was so new that the sequence was only released maybe 2 weeks before this, on 10th of January. So for our first case, of course, we do WGS just to make sure that really is it SARS-CoV-2, and is it and we found it's identical to the one reported from Wuhan. So it's double, triple. You know, we are saying we like to double confirm everything when we our first cases, right? Yeah, and then we had a few outbreaks and all that. You know, here we use, obviously epidemiology is still important. So when we found that the first outbreak we had was from a visitor who went to a Chinese New Year dinner, and then they also went to the church, and people who attended dinner went to another church. So So actually these two church outbreaks were actually related to the person who went to a Chinese New Year dinner, right? So of course we could figure that out by epidemiology, right? But we also confirm with sequencing that it is the same link. These classes are linked and linked to the visitor from Wuhan, right? So another reminder that genome sequencing must always go with the epidemiology, right? The sequence can't tell you how it spread. You need the epidemiology, right? And in this case, this is how it spread. This was a Chinese New Year gathering here. So so they went to the church before, then went to Chinese New Year, and then in the New Year gathering, they went to another church. So it's all linked, right? And they started from two travelers who arrived from Wuhan, yeah. Shortly before, now our first case, remember, was 23rd of January, but unknown to us, the travelers were already carrying the virus and not detected yet, right? So we need to diagnose it later on, right? Yeah. Then the other thing, as we started to sequence the viruses, whichever ones we had positive, one day we found something very strange. In fact, the laboratory asked, was there something wrong? They went to sequence again because we found that, the virus has different, what I call open reading frames and coding for different genes. And we found one of the open reading frames, open reading frame 8, a few hundred bases disappeared, about, and then we in the end we found that it wasn't a laboratory error, that actually that virus had a missing segment, and this had been observed before with the old SARS, when researchers found that this has been linked to maybe more severe disease and all that, So in our case, we actually found these cases in Singapore, so continuing monitoring, we find that this case was not reported anywhere in the world. Later on, we did find one case reported from Taiwan, but otherwise it appears to be in Singapore. So what's the impact if it's missing? Does it get worse or better? So our clinical data now, when we tie in together with who had this deletion in the genome, we found that these cases tended to do slightly better. They did not go to the ICU or what. So it's not very strong evidence, but it seemed that the deletion made it less severe, right? But later on, the deletion disappeared. So it meant that our control strategy was so good that even this chain of transmission stopped, right? So this virus disappeared, right? And the sequencing showed that because it disappeared, it shows that our control was quite effective. We basically interrupted transmission, right? So this was, we didn't set out to do that, but these are some of the things when you're doing sequencing, you suddenly find out these are it's not so incidental. It is incidental in the sense that we did not look out for it, but as you sequence the genome, sometimes you have surprising findings, right? Later on, I'll show you another surprising finding we had, right? And then we had the dormitories with our foreign workers, and we also link epidemiologically to a place they go to every weekend where they go shopping, right? Many of the foreign workers go to the same place to shop, right? And the majority of these foreign workers are from like India and Bangladesh, of course some from a few other countries, but Bangladesh and India are the main ones, right? And so our sequence show us it's not just B6, right? That we find that all these B6 were almost identical, which means that it is not like we have say 20 dormitories. It's not 20 dormitories with different sequences, So whatever's happening, whether they got it at the what we call Serangoon Road, or whether at the workplace they started to spread to each other, we are not quite sure. But whatever it is, it was the same strain spreading to all the dormitories, right? So it helped us to understand clearly what the pattern of spread was, that it wasn't due to multiple strain, but it's the same transmission event which was happening. And the other thing is that B6 was not described from Europe or from US. B6 was described largely from the Indian subcontinent, right? So even early, in fact, at the time it did not have a clade name, so we call it the O clade. O stands for actually others, right? It's not one of the main Western clade, so it's quite clearly from from the workers and linked to the homeland, right? And all B6, right? And then we also know, and then of course during this time, off and on, we do have some local cases, right? And local case, we want to know, is it leaking from the dormitory outbreak, or are these new imported cases? Because we still have people flying in from various countries, right? Right. So we use the surveillance to find out when we have local cases, community cases, whether it is due to the dormitories or travel. And actually, a few of cases we did find were from dormitories. Then we try to find out what was your contact with the dormitories or with the foreign workers and all that. Because what we did was that we rehoused all the foreign workers into separate housing, isolated them, and we separated the infected from non-infected, right? So it was a huge operation. We brought in the armed forces to help to do that. We actually opened some army camps in other places so that they were housed there. And actually this showed that this strategy was quite effective, because not only they did bring down the cases among the dormitories, it also prevented the dormitory cases from causing community outbreaks, in the local community. Right. And then this was news, and actually it spreads again from Europe, the 614G, and it became very common. And then some countries, not just Malaysia, but some countries say that, this is a new thing. Maybe it's a cause of the increased transmission and all that. I think also there was some statement from Hong Kong and other places saying, is this mutation 614G causing increased transmission? I like to warn that it's not easy to come to this conclusion, because it may be what you call founder effect, because wherever transmission occurs, naturally over time there will be a predominant type of, or in this case, a predominant mutation, right? 614G in the end became the dominant mutation all over the world, right? But it does not mean that by itself it caused the increased transmission. Increased transmission can be just due to inadequate quarantine or whatever, nothing to do with change in the virus, right? So whether when you have increased numbers, whether it's due to change in virus, I think we have to be careful about blaming the virus, right? Because sometimes normal measures happen. And in fact, for Singapore, we saw 614G, but we are reluctant to say our cases were due to that. And actually, whatever 614G entered to us from the UK and wherever, we by strict quarantine and isolation, we stop transmission. So our point is that with good or standard public health measures, we could actually contain the 614G. I won't say quite easily. It took some effort, but it's not as if you can't control it by normal public health measures. So we could, right? And then this was our second surprise. So we had not much local cases. We say, okay, we have opened up to more travelers, and then we're getting more travelers infected, and say, okay, maybe what we'll do is we need to find out travelers who are positive, what is their genotype. So we did surveillance on travelers, because those were the only cases we had, so that if something happened, then we know which travelers would spread it. Then we also know which genotypes are from which countries or which continents, because in some countries there was not much sequencing going on, or it was going on in December. Maybe they take 1 month, 2 months to upload the data, right? So our purpose of sequencing these travelers was just to see what's coming out from different countries, especially from the region. And then we found, again, a surprise by accident. It was meant for travelers. Then we found that travelers who came from different countries and were not related to each other, how come they were having identical genotype, right? So again, we had to check for laboratory error, right? And then in the end, we found that actually what happened was there were travelers coming in, they were actually COVID-free, negative, and then there was transmission going on as during the quarantine period in the hotel. So it was spread in the hotel itself. It was not the travelers being infected, right? And of course it's another story, but we later tried to find out how they stay in their own room, how could they transmit to each other, right? And actually in the end we found, although I showed one hotel, I'm being unfair, we also found it in 1 or 2 other quarantine hotels. So we know that spread in quarantine hotels is a problem, even though we think that they stay in their own rooms and never go out, right? So that was a surprising finding. So sometimes you do surveillance, you have surprising findings, right? And of course, from December onwards, first the UK and later WHO started to have this classification. So variants of concern, VOC, it implies those which have quite good evidence that either they are transmitted easily or they cause more severe disease, or they have antigenic change so that the vaccine may not be so effective. So from January onwards, we said we'll sequence every case, right? So when your numbers are low, you can sequence every case. Of course, those cases where the viral load is low, our sequence not very successful. So maybe half our cases could be sequenced, but we sequence everyone, right? To see whether import any variant of concern, right? Or even variant of interest, right? And we wanted to have a very fast turnaround time, because no point knowing it 1 month later, right? We wanted to know it within a week. And sometimes, in fact, our bosses say, can I have it in 3 days? We say, cannot lah, 3 days cannot, yeah. So we promised 10 days, 14 days, and then in May, right? I remember I was in hospital. The first outbreak we had was actually in our hospital, in which fully vaccinated staff in the hospital were infected by this new genotype, and that was when I realized that even with our very good efforts, what we think effective control efforts, both in the hospital as well as in the community, what we are doing was seeing a new type which seemed to be very transmissible. Previously, when we just quarantine the household contacts, that's fine. Then we stop the transmission. Now we quarantine the household contact, they already spread to other people, second generation. So we embark on more stringent control after that. So we say, okay, even your work contacts, now we need to quarantine you. So lots of quarantining happening in May, right, to contain Delta. And in fact, we showed that initially we did manage to control Delta. In fact, the initial one linked to Changi Airport, a few hundred cases, they spread to shopping centers. Eventually, we actually did close it down. And how do we know that we were successful? Because when we started when we did our whole genome sequencing of all, at the time we were doing every virus, every specimen, we find that even later on when the other Delta coming in, right, the original Delta from the airport have disappeared, right? We have controlled it. It's no more circulating the airport strain. It doesn't circulate anymore. So we know that although there was a lot of effort, we did manage to control the initial large outbreaks on the airport. So even if Delta, with a lot of effort, it was possible, right? It was possible, right? And initially, so many cases, we said this unexplained. So initially, when we had a shopping center quite far away from airport, oh, where did the shopping center cases come from? Right, we can't explain, but then we managed to link by genotype but this still belongs to an airport cluster. Then we link that, oh, this person traveled from here, met somebody there, and the relative went to the shopping center, and then the shopping center staff, this was the common factor spread to everyone who visited that shop. So we managed to explain the unlinked cases, and we help to link them. So sometimes we get the clue from the WGS. And then the field epidemiologists went back to interview and then to find what the link was, right? Yeah. So these are our Changi Airport cluster, right? And in fact, our first case, one of our hospital staff, the husband or something, was working at the airport as an officer, right? So was it due to that same thing? But later we found that it was different. It was not due to that. So what the staff had was a different gene sequence from the one in the hospital, right? Yeah. So our ministry tried to excuse and say, okay, we have so many cases because some safety measures breached, and it's not possible to seal it ourselves off totally. Actually, I say no country, only one country can do so, and you know which country it is now, so China is sealing themselves very completely, so that's why they have very few cases. Yeah. So as I said, the Changi Airport one spread to the shopping centers all over, right? And so these are all taken from a newspaper. Whole genome sequencing by publisher revealed that the visitors to GEM and Westgate, different shopping center, have similar sequences, and we found that often they're the same sequence to the cleaner. So one other risk group, we actually found one of the risk groups we found were actually the cleaners at the various shopping centers and markets, right? And they are at the risk. So when we did our, and because some of the cleaners, they also work in the hotels with quarantine cases, they get it from the quarantine people who are positive, right? So it's an issue, but so we definitely targeted cleaners for our vaccine once they were available, right? Make sure they're vaccinated and test them regularly, right? And then later it's spread to Bukit Maravu. This is a hawker center. So this also related to Changi Airport case. So some people from the classes from the shopping centers went to visit. You know, this where most people go for eating, for marketing. So the place where you have the market and the place we eat our food is roughly the same area, separate. They are separated but same area, right? So this was linked to this place, and this case actually quite near MOH, you know, by the way, yeah. And one of the reasons was that many of the elderly, despite whatever we advise them, the elderly like to sit and talk and meet together, you know, when they can. Of course, this normal life, and that's a good way to live your life, you know, to have social interaction. But unfortunately, many of the cases then spread among these elderly, and of course, with the elderly, we started to see more fatal cases which we never saw for the past year or so, right? And so yeah, so we trace that back, right? And then we also found some in the housing estates. You know, we have many housing estates. Maybe 90% of Singapore people stay in housing estates, right? And we found interesting thing that in these housing estates, we find that those staying on the second floor can spread to 5th floor, 7th floor, all on the same vertical block, right? Very strange, right? And then it's less likely for the one which is on the top block to spread to the lower block. We up to now we still wondering how it is, but so we proved that because they are identical, they were not. So the question is that because a lot of people stay here, do they all maybe go to different markets or shopping centers and get infected there? No, we find that some of these sequences are actually unique to these blocks, and then it's not like those from here will spread to here. Many cases appears to be all from the same, not just from the same block, but from the same so-called column. You know, so we know that there was something happening within the housing estate, and so then they have to question that. Is it because they meet in the lift? Is it because they take the same bus and all that? So that's another story, you know. It's not fully solved yet. Or is there some ventilation problem? So we actually look into ventilation in the buildings as well, right? So even when you have community outbreak, the whole genome sequencing help to show that actually it's spreading among the residents and not because they all go to the markets and get infected in the markets, right? Right. So that
And we
a lot of
have
effort
2 minutes
Yep.
more, Raymond.
Okay. And then sorry, was there something? Yeah.
2 minutes, 3 minutes at max.
Oh, okay, sorry. And then we have
Thank
one
you.
link to the fisheries and then spread to different markets, right? So what we do for sequencing now, we also check that when the sequence is different, we can show that, oh, this is a reinfection case. Because those infected last year, no Delta, right? But now we have Delta, so we know it's a reinfection, right? So now our new WGS strategy, we have so many cases, we don't do all anymore. So we end up doing about 1 to 2% of cases at the moment, right? And these are priorities. We now settle on travelers. As we open up, we want to do more travelers. When we have classes of outbreaks of interest, when we have special links, only special link, and MOH will tell us, okay, this link, we want to find out if it's hospital transmitting it. Then we will, for example, then we'll investigate it. And then we try to go through our ILI or sentinel surveillance and see whether we can use that to do sentinel sequencing, right? And then the other reason is to use the variant to see if we've got a really new variant, AY23 or whatever, to help in our variant surveillance. Okay, so and then we need to tie with our other people to assess the variants, right? And this just to show okay, this is how it has changed the different variant types, but I give you, and then we also model it on GIS8 to see which part of the virus it affects. And then our last slide, so this shows our latest data from September to November. We have about 3,000 genomes, and this our main one is either 23 or the new one is 23.1. So the 23 we added some added mutation, become 23.1. So currently this is what we have at the moment. So thank you.
Thank you, Dr. Lin. It was very, very, very nice. I mean, it's like a time lapse. You see it from the very first until the very now, and what is happening, and the waves and the either new infection or reinfection. I think it's something that is very interesting to pinpoint, I guess, and also now that a lot of people are already vaccinated and all. So I guess that's also another factor that we should keep an eye on about vaccinations among the people, right? Okay. Dr. Raymond, we have to move on to the second speaker, Dr. Dirk from the Netherlands. So it would be very nice if they can join along the way until the third speaker. But now I would like to invite Dr. Dirk. Thank you again, Dr. Raymond Lin.
Thank you.
Very, very nice presentation. Dr. Dirk, you are already in?
Good morning. Can you hear me?
Yes. Goedemorgen.
Goedemorgen. Yes, it's quite early for me. So,
I guess so.
But I'm very happy to be here, and it's always nice to join for a meeting like this. And I really hope we can meet in person again in the near future. But till that time, I'm just happy to join that I can join via the virtual meeting like this. So,
okay.
Yeah.
Thank you. So you are from RIVM. Earlier this morning, I already read your excellent CV. So you are from RIVM.
Yes, Dutch Public Health Institute.
Yes, I have visited there. So
great.
because I was doing my PhD with Tom Ottenhoff in Leiden on tuberculosis. So
fantastic.
Yeah. Okay. I guess you will be talking about surveillance of SARS-CoV-2 and other respiratory pathogens in the Netherlands. That was kind of the topic that was in the right.
Yes, I will be mostly talking about SARS-CoV-2, and we'll see how fast the presentation will go if I'm able to touch on the other respiratory pathogens, but we'll see where we get. Shall I start sharing my screen?
Yep. You have time available is around 28 until 30 minutes. So time and floor is yours.
Yes, thank you very much. So can you all see my screen?
Yes, very nice.
Okay. So you can see the slides, right? Yeah, I can see it now. So as this is the beautiful building of the RIVM, the Dutch Public Health Institute, and I'm happy to be able to talk today about the national SARS-CoV-2 surveillance structure in the Netherlands this morning. And I would like to talk about the surveillance of genetic changes within SARS-CoV-2. Unfortunately, I missed a large part of the previous talk, so I don't know if there's much overlap, but I think there are always different strategies within different countries. So I think it's very nice to be able to compare those here. So I want to shortly discuss when and why are we doing SARS-CoV-2 sequencing in the Netherlands. And when I talk about sequencing, I'm talking about whole genome sequencing, of course. What is the setup of a national surveillance program? And I want to obviously talk about the detection of these variants of concern, how they have been detected, how they were introduced in the Netherlands and further spread. And I want to touch on how much should one sequence. And together with the European Centre of Disease Control, we have developed a strategy to discuss numbers and decide on how much should one sequence to get the good information without spending too much money on it, which is because whole genome sequencing is, of course, a very expensive technique, although it will provide a lot of valuable insights. And if time allows, I want to talk about the surveillance of other respiratory pathogens. We tend to forget that there are lots of other respiratory pathogens around that are also important for pressure on the hospitals, etc. So surveillance for those pathogens remains as important as it always has been. And then we have 3 levels of surveillance currently in place that I would like to touch on if time allows. So talking about SARS-CoV-2 sequences in the Netherlands, when do we perform SARS-CoV-2 whole genome sequencing? We use this in source and contact tracing, especially if there's a specific travel history, for example, to areas around the world where specific variants circulate. And when there are outbreaks with an unusual signature, so if you have a large outbreak, for example, in an elderly home or in schools, then we can see with using sequencing how many introductions were there, how many, was it like a super spreader event, or were there multiple introductions to cause that outbreak, etc. And of course, we do sequencing when there are special cases, either with a special phenotype, severity, or when novel variants are introduced in the Netherlands. We want to sequence and see if we can track that. And which is the main topic of today, we perform sequencing as part of the national surveillance in the Netherlands. And why are we doing that surveillance? It's good to realize the rationale behind that. And when I talk about national surveillance, I'm talking about a random selection, and that random selection of samples is very important, of course, because you want a trustworthy reflection of what is actually circulating in the country. We have way too many cases to sequence all of them. And so therefore, the selection on how you do that selection is very important. What are the main aims for the surveillance? And that's baseline data. So what is circulating, where is that circulating, and when was it introduced? And by that, we can monitor the genetic stability of the virus. How much is it changing, and which mutations are appearing in functional domains? But was already touched on by the previous speaker, but of course it's very important to use it as a vaccination follow-up on a population. Are there any clues for vaccine escape? Like we see that, for example, for the influenza virus, where an annual update of the vaccine is required because of this vaccine escape. So we're keeping track of that. And we're not only doing that in the Netherlands, of course, because you need to place that into an international and worldwide perspective. And a very important thing also in the light of today's meeting is that we use this presence of all the variants and variants of concern in our modeling, and especially the modeling of the epidemiologist for the prediction of number of cases. If we know that a certain variant of concern has another reproduction number, then of course that will influence the predicted number of cases in the Netherlands, and therefore the variants circulating in the Netherlands are very important to know. And that is related to what we call the surveillance for action, is that we might have different measures so by the government because of the variants that are circulating. Of course, it's good to keep track of the general epidemiology of the virus. So how does the virus spread, and the previously mentioned outbreaks. And although not as touched on as much as the other reasons for sequencing here, is that of course you need to keep track on your diagnostics. Do we see any changes in areas that we know are important for example, for the diagnostic primers? This sequencing is part of an extensive surveillance toolbox, because the sequencing is important, but of course the epidemiological parameters are as important, because it's the combination on the epidemiological data and the sequencing data about the virus that give you insight in this pandemic. And another thing that I'd like to stress out here, that although it is very important to know how the combination of variants that circulate in a country are present, however, currently in the Netherlands, we do not have any different control measures for the different variants. We have the lockdown is not different if at this point not different if a certain variant is circulating or another variant. So how is the national surveillance being set up? Currently, we have over 30 participating laboratories, and these laboratories come from healthcare facilities like hospitals and the test lanes. When I talk about test lanes, that is that's the community testing. As soon as you have symptoms, you can get tested in the Netherlands. It's very easy to make an appointment, and we have all these test lanes. Often you just go there by car, and they swap you from out of your car. So that is very important to include those in the surveillance, as that will give you an idea on the general circulating of variants. And this is a nationwide network of laboratories. And currently we are sequencing around 1,500 samples per week. So that's a minimum of 100 to 200 daily. And I'll touch on that number and how we came up with that absolute number, because these 1,500 are independent of the incidence. So independent of the number of cases in the country at that point. So if there's a peak, we still have around 1,500 sequences a week. As previously mentioned, it's a random sample, and that is very important, that we really try to avoid any biases in clusters or outbreaks. Sequencing is performed at the Center of Infection Prevention at the RIVM, but in combination with 12 additional labs. We call that the SecNet, the Sequence Netherlands network, that have been established during this pandemic to increase the capacity for this sequencing. This sequencing allows detailed reports to policymakers, the outbreak management team, the government, of course, and we report back to the laboratories and the municipal health services. We deposit all sequences in GISAID on a weekly basis to make sure that we share the latest information from our country with the world. So it has been probably mentioned earlier today, but it's very important to keep an eye on these so-called variants of concern or variants of interest. That's the stage before the variant of concern, when there's not too much data yet. But a variant of concern, just to remind everybody, is a variant of the SARS-CoV-2 virus that has been demonstrated to be associated with either an increase in transmissibility and thereby the epidemiology, or an increase in virulence and clinical presentation. And it might also be involved in a decrease in effectiveness of the measures, like the 1.5 meter distance, for example, or the available diagnostics and vaccines or therapeutics. And especially, of course, the vaccines is worrisome if there is an indication that there's a diminished sensitivity. So to show that it is indeed very important to measure this, and what you can measure with this surveillance, is that variants of concern are often characterized by substitutions in functional domains of the spike protein. The spike protein is responsible for attachment to the target cell and subsequent entry of the virus. And what we often see in these variants of concern is that there are in all three of the functional domains of the spike protein, we see substitutions. For example, in the receptor binding domain, that can result in improved receptor binding and replication. Thereby, you can have increased transmission and spread. Changes in the receptor binding domain might result in antibody escape and therefore a change in vaccine efficacy. The N-terminal domain, depicted in purple in the structures on the bottom of the slide, we don't know exactly all the functions of the N-terminal domain, but we know it's a very important target for neutralizing antibodies, and therefore any changes in the N-terminal domain can result in antibody escape and thereby diminished vaccine efficacy. And the third important functional domain of the spike protein is the S1, S2 region that need to be cleaved to activate the protein, and changes in that domain can alter furin cleavage and replication and infectivity, and thereby changing and increasing transmission and spread. I want to give a short example of a study that the RIVM performed together with the Amsterdam UMC, previously called the AMC Hospital. And what you see here is we investigated seroreactivity against the three variants of concern at the time, so the alpha, beta, and gamma variant of concern, and we took sera from either infected individuals or vaccinated individuals. And if you compare the two figures at the bottom, you can see the reactivity of the 3 variants of concern, compared to the original wild-type strain as first identified in Wuhan, you can see that there is a diminished binding titer, and that effect is strongest for the gamma variant, depicted in the bottom left panel in green, and you see that reduced reactivity in both sera from infected and vaccinated individuals. And you can see that it's not only the binding, but also the neutralization of the virus is affected. But interestingly enough, there we see a bigger effect for the beta variant compared to the gamma variant. So it is important to monitor these changes in the spike protein and in the virus in general, because as you can see clearly in these examples, these changes can result in reduced sensitivity for neutralizing antibodies and thereby possibly vaccine efficacy. So I want to show you an example of variant detection within the Netherlands, and the next few slides will be to give you a general idea of circulating strains over time with geographical distribution, and of course that involves the detection of variants of concern. And in the figure on the right, you can see that over the months we have gotten a very nice general idea on variants as they circulate over the Netherlands and within a good geographical spread. So we take a little step back in time. It was late 2020 or early this year that Alpha variant, as it was first detected in the UK, or the B117 lineage, it was detected, and we had to introduce them. We had to monitor the introduction and the spread of this virus. And it's depicted in these visualizations that we made using NextStrain. And what you can see here is it's a picture of the Netherlands, and with every panel represents a week, and it starts late previous year. And first we had the EU1 and EU2 lineages. They were very much similar to the original Wuhan strain. And what you can see that late 2020, you see like an orange part in the pie diagrams over the whole of the Netherlands. And then with the months, if you look at the bottom 3 panels, you see that that share in all the variants detected, see increasing, and the orange part is becoming bigger as a part of the of all the variants circulating. And especially if you go further in time, you suddenly see a very steep increase in this particular variant, and until it hits close to 100%, as you can see as of week 10 of this year. So the Alpha variant, it was first detected in November and December in 2021. 2020, of course, that needs to be. And then we saw a rapid increase in the earlier this year, and it became the majority in all 12 provinces of the Netherlands mid to late February, so around week 7 of this year. And in March, it was about 90% of all cases in all provinces. And we were very detailed. We were able to monitor that in very much in detail. And after it became the most prevalent strain in the Netherlands, it remained the most dominant variant for several months. And in that same time, there was also the beta and gamma variant of concern circulating, and those remained low. We did detect them in the country, but it didn't really take off. And so this is an example of how a certain variant is introduced, we monitor it, and then suddenly you see a rapid increase. And the beta and the gamma variant are the opposite. For example, if we look at the gamma variant, you can see in the left panel that it is observed in multiple areas in the Netherlands. But if you look at the phylogenetic analysis on the right, you can see that there are distinct clusters, and all these clusters died out. So there was some local spread, and all these local clusters were unrelated. So there were multiple introductions in the country, and then this virus variant died out, and there was no structural increase in prevalence. And we were very well able to monitor that with high resolution. Then obviously Delta came. The Delta variant first detected in India, but after a while, it spread around the world, and we were quite early able to detect that variant in the Netherlands. So especially around April, we saw the first cases in the Netherlands, and the first cases known to the Dutch Public Health Institute mostly had to do with travel history. So there was also extra sequencing for that when people had a positive test and it was known that they had traveled to a certain area in the world where this variant was circulating. First of all, that was India, but later there was a lot of the Delta variant also circulating in, for example, the United Kingdom. So when people came back to the Netherlands and they got a positive PCR test, we performed sequencing and we were able to detect the Delta variant. That was specific cases that we sequenced, but also soon after April, we saw incidental cases in the random sampling set. However, there was some spreading around the Netherlands, but it took until June until the prevalence really started to increase. And that is shown here in this bar graph. You can see the Alpha variant in blue, and you see the Delta variant in green. And in April, May, suddenly the Alpha variant started to diminish, and the share, the proportion of Delta variant very rapidly increased in the Netherlands. And that coincided with a less strict measure at the time, because it was going rather well with SARS-CoV-2 cases in the Netherlands. So the country was opening up again, and that resulted in a very rapid increase in number of Delta cases in the Netherlands. So after showing you the data for the surveillance and giving some examples of that surveillance, it's very important to think about how much should we sequence, because of course the more the better. The more sequences you're able to obtain, the more information you have. But can you give a number to that? How much should we sequence? And for that, two levels of power or numbers are important. And the first one is, when are you able to detect the first positive sample of a novel variant? So let's call that variant X. So is the variant present or absent? So in the case of the Delta that I just mentioned, we were already able to detect it in April, but how much should you sequence to detect early? And another important power that you need to consider when you think about numbers of samples that you should sequence is that when are you able to estimate the increase over time and estimate that with high confidence, of course. And why is this important? Because it helps to predict the total number of cases. As I previously mentioned, depending on which variant is circulating in the country, the reproduction number of the virus might differ, and therefore the prediction and the models about how the pandemic will evolve might change. And on the panel on the right, you see in the different green colors how the sequencing was performed earlier this year in Europe, and you can see that many countries are sequencing over 500 specimens a week. How did we get to the 1500? Because it is as I said, it is important to sequence a lot of specimens, but it is expensive and it requires lab capacity, and that includes both the reagents, personnel, and of course equipment. And together with ECDC, we came to the number of 1500, and it is given in this table, and I've picked out the most important lines of this table at the bottom, that it was calculated that if you sequence around 500 samples a week, you are able to detect a variant with a prevalence of around 2.5%. If you sequence around 1500 samples a week, you're able to detect a variant that is circulating with a prevalence of around 1%. And if you double that, so you go to 3000 samples a week, you're only able to detect variants that are 0.5%. So with doubling the number of samples, you only go from 1% to 0.5%, which sounds logical, but it is a lot of extra work for little extra information. And again, it is important to have that power to use for the detection and the presence of variants of concern. And so we, within the RIVM, all based on our own data and on international data, we calculated what day are we first able to detect a new variant of concern, and how many days does it take if you consider a variant just like the alpha variant with an increased transmissibility. How many days do you have till it hits 50% prevalence in your country? Because that kind of gives a time window that you can use to change that behavior of the virus and behavior of the people, of course, and thereby hopefully dampening the number of cases. So that time frame is really important. And when are you reliably able to predict exactly when it will hit that 50%? And of course, there's always a margin in that. And with our own calculations, so previous slides were the calculations from ECDC, so the European Center for Disease Control. And here I show you our own calculations. And the red arrow indicates if you sequence around 200 samples every day, then approximately 60 days, like 58 days, so that is around 2 months, you have till it hits that 50% prevalence. So till 50% of all cases in your country are that novel variant. And the current prevalence at the time that you first detect it is around 0.5%, and that corresponds to the 0.5 to 1% in the previous slide. If you go from 200 samples a day to 300 samples a day, it almost sounds like it is no extra work. 200, 300, but of course it is extra work. It is extra reagents, extra capacity in the lab, personnel, and you only gain a couple of days from 58 to around 63, according to these models, till it hits that 50% prevalence. And so it's a lot of extra work for limited extra insights, in our opinion. So therefore, we sequence around 200 specimens a day, which results in around 14 to 1,500 samples a week. And this model and data is showing that it worked in case of the Delta variant, and it also worked for the Alpha variant at the time. But what you see here in the 3 different colors, you see the prevalence of the most important variants of concern. So in red, you see the steep rise of the Alpha variant as of January, and in green, you see the Delta variant. And if you look at the 4 different panels, so if we look to the top left and top right panel, you can see that there is very limited data, and so the dots indicate the data points, and then you see the modeled increase in prevalence, and with the shade you see the confidence interval. And although there's only minimal data known in the top left panel, you can see that it gives a rather trustworthy prediction on how that variant will increase in prevalence. And the more data becomes available over time, you go to the bottom panels, bottom left first and then bottom right, you can see that the confidence interval narrows, and it does indeed follow that prediction. The curve, and when it hits 50% prevalence, is only shifted 1 or 2 weeks, despite the fact that that model was made with very little information as present in the top left panel. So indeed, with our current sequencing numbers, we are able to make these kind of models with high certainty and confidence. So surveillance was able to detect the first cases of Delta in the Netherlands over 2 months, so over those 60 days before it actually hit 50% prevalence. And we had the capability to predict the increase in prevalence and a moment that the virus become the most abundant variant. So to conclude, we have a strong and sustainable SARS-CoV-2 surveillance where the Dutch Public Health Institute plays an important role, but we have a very valuable network of laboratories providing the samples and helping us doing the sequencing. And we're able to detect the introduction and spread of variants of concern and variants of interest. And for that, we use around 200 samples a day. And that allows us to first detect new novel variants of concern or any novel variant 2 months before. And we have a good curve fit to predict the increase in prevalence. And this is important to predict the number of cases and hospitalizations. And that, of course, that information is used by our outbreak management team and our government to decide on the policies and measures that are needed in the country. So I think I do have a little bit of time left, so I can shortly touch on the surveillance of other respiratory pathogens, because there are other respiratory pathogens still out there. And although we didn't see much influenza virus in the past year and a half, influenza is back, and we were able to detect it. And we have 3 levels of surveillance, and we have the family doctors, so the GPs, we have the weekly virological records, mostly from hospital laboratories, and of course we have this community testing for SARS-CoV-2 in the test lanes that are previously described, and that is also a very valuable source of information. And why is this important? Why should we monitor these other respiratory pathogens? It's important to know what is circulating and what viruses do cause these symptoms, like that you also can see for SARS-CoV-2, and that way it helps to predict how many people will have symptoms and will get tested. So it is also important to predict your testing capacity in the Netherlands. And especially influenza virus and RSV, of course, can cause hospitalization, which will give even more pressure on the hospitals. So this pyramid kind of describes how the testing in the Netherlands works. And the bottom one are just individuals with respiratory symptoms within the community, like not too severe. Then we have people that actually have more severe symptoms, and they go to the test lanes, so they have the community testing. Then the next level, they might go to their family doctor, so the general practitioner, and eventually they might end up in a hospital. And so the higher up in the pyramid, the more severe the disease, but also the more time it takes from the very first onset of symptoms until the diagnosis and the testing and getting an idea on which virus is circulating. And today I want to give an example in the next few slides.
Excuse me, Dirk.
Yeah.
Can we have a kind of a wrap up in 1 or 2 minutes?
Yes,
maximum?
I will. Yeah, so I will just show you how we are testing here then. That is that we have test samples, a random selection from our family doctors and from our test lanes. And what you can see here is that influenza virus did disappear, but as of this past month, influenza virus is coming back. So within a random selection of samples from our test lanes, and it's a rather small selection, it's only 5 samples per week, we test a broad panel of respiratory viruses. And although it's mostly rhinovirus and SARS-CoV-2, of course, there is low-level detection of influenza virus and RSV, and we are able to monitor that, and that helps us in predicting the pressure on our hospitals. So with that, I'd like to end. Thank you very much for your attention. And again, I would like to thank all collaborating laboratories and institutes in the Netherlands.
Thank you so much for very interesting talk. I guess, Dirk, the importance of, how do you say it, the lay community surveillance, and also not only trying to find out the SARS-CoV-2, but it's also trying to find others potential microorganism which might cause burden to the hospital. So I guess exactly. That was a very, very important message on your talk. So I will have there's a question in the Q&A, but I think I have to move on to the third speaker. Is Dr. Adam here already in the room? Dr. Roberts? Adam Roberts?
Yes, good morning.
Hey, good morning. What time is it in Liverpool?
It is 3:56 in the morning.
We're sorry.
That's okay. It's a pleasure to be here.
Okay, it's great to have you here. So earlier this morning, I already introduced you with your CV, impressive CV. So I think we can proceed to your discussion or your talk. The floor and time is yours on the applications of whole genome sequencing to strengthen surveillance and detections of antimicrobial resistance. That was the topic that was written in the agenda. So time and floor is yours. 30 minutes.
Okay, so thank you very much to the organisers for giving me the opportunity to take part in this webinar. It's an absolute honour and a pleasure. I will just start my screen. So I'm assuming that you can all see the correct screen there. So there we go. So as mentioned, my title is the application of whole genome sequencing to strengthen surveillance and detection of antimicrobial resistance. And within this seminar, I really want to give an overview of the benefits of incorporating whole genome sequencing and subsequent analysis on top of antimicrobial sensitivity testing when thinking about susceptibility to antibiotics commonly used in different countries. So the outline of my presentation will be in 5 broad parts. I'm going to give an introduction to AMR first. I realise that we are changing from SARS-CoV-2 and COVID disease into AMR, so I think that will bring us into the correct space. And then I'm going to just discuss the data insights and the translational capabilities and values of using whole genome sequencing in addition to antimicrobial sensitivity testing. We can't have a discussion of whole genome sequencing in bacteria without also thinking about GLASS, the Global Antimicrobial Susceptibility Surveillance System. So we'll have a look at that as well. And then I'm going to give you some examples of fledgling analysis collaborations that I'm involved with, and also some more mature ones. And then we'll finally discuss a roadmap to implementation of such whole genome sequencing capabilities. So let's think about first, what is AMR? So antimicrobial resistance is the resistance of a microbe, and that's a bacteria or a fungi or indeed a virus, to an antimicrobial agent, in the case of bacteria, an antibiotic, to which it was originally sensitive. So it's a change. It's not discussing the inherent resistance, which is present in some bacteria. What it means for humans is that infections that cannot be treated with antibiotics as easily as they could before. And I have an example here, posted with permission from my young daughter. This is her shin. She got an infection just whilst playing in the forest. That wouldn't respond to antibiotics, and in the end it had to be operated on to remove the offending tissue before osteomyelitis occurred. So this just illustrates that a bacterial infection can happen anywhere to healthy individuals, and sometimes they are resistant to antibiotics. So how does it occur? Well, it can occur either by mutation in the replicating DNA strands, as we can see here on the left panel, or it can occur by acquisition of DNA by a horizontal gene transfer. And when I'm talking about horizontal gene transfer, I'm talking about transformation, which is the uptake of naked DNA, usually released by dead cells, by a competent recipient cell. We also have conjugation of both plasmids and transposons from live donor cells into a recipient cell. And we have the transfer of DNA by bacteriophages, bacterial viruses, which can package some of the original host DNA in their phage heads, as we can see here, and then inject it into a new recipient. And if any of this DNA contains genes responsible for antimicrobial resistance, you can get a stepwise evolutionary step, which can give resistance to one or indeed multiple antibiotics in the case of acquisition of, for example, large plasmids containing multiple antibiotic resistance genes. So what are the drivers of AMR? Surely we can just have more and more antibiotics, so when resistance occurs, we can use a different antibiotic. Well, as we all know, that's not the case, otherwise we wouldn't have AMR. So let's just go through some of the drivers. So first off, we have limited targets of antibiotics, and we have multiple resistance mechanisms. So this cartoon here really just outlines the main targets of the various antibiotics that we use: the cell wall, nucleic acid synthesis, metabolic pathways such as folate synthesis is a target, the cell membrane and protein synthesis. And all of the antibiotics that we use target these small number of processes. And then if we think about in terms of antibiotic resistance, we have multiple mechanisms of resistance, and often multiple mechanisms of resistance can provide resistance to the same antibiotic. So there's multiple ways a cell can overcome the effect of a particular compound. Next, we've got a discovery void. So if we think about when antibiotics were first introduced to market, we can see that in the sort of the 1950s and 60s, that was a golden era of antibiotic discovery. All researchers needed to do really was to grow some bacteria from soil or from moldy fruit, and they would find a microbe producing a novel antibiotic, and they could bring it to market relatively rapidly. Since the 1990s, there have been no new class of antibiotic brought to market. So we're in this discovery void, which means that we are continually relying on the older antibiotics that we've got in our formulary. Now, an additional point I want to make here is that our reliance on antibiotics as a society goes back way beyond the introduction of antibiotics in industrial quantities. So if we just think about sort of the traditional uses of antimicrobial practices, if you like, we've got examples such as in Greece and Serbia, moldy bread was traditionally used to treat wounds and infections. Now, if we think about the mold here, this is probably Penicillium notatum or Penicillium chrysogenum. Now, both of those are the original producers of penicillin. So we can see why there may be some antimicrobial effect within that traditional practice. Similarly, with warm soil, which has been used in Russia to cure infected wounds, many of the antibiotics we have are produced naturally by streptomyces, which inhabit the soil. So again, you can see why warming it up and increasing the metabolic activity of the microbiome within the soil, you get the production of antibiotics. This is a more obscure one. Babylonian doctors many, many years ago healed the eyes using a mixture of frog bile and sour milk. Now that sounds fanciful, but actually frog bile is made up of quite a number of antimicrobial bile acids, and sour milk has probably turned sour due to an overgrowth of Lactococcus species, which are also natural producers of antibiotics. And then finally, to keep milk from turning sour, Russian residents used to put a frog in the bucket of milk. And again, this sounds crazy, but actually there's been some work done on this, and the skin peptidome of the Russian brown frog, Rana temporaria, actually produces multiple peptides called bradykinins, and these are antimicrobial in nature. So again, we have this element of truth and scientific basis for use of traditional medicines. And this has also recently been shown by analysis of traditional medicinal plants in Indonesia. There are at least 2 with really good efficacy against Candida species. So we can see here that traditional medicinal plants in Indonesia may well be a source of suitable pharmaceuticals to develop antifungals. So let's just recap on the drivers of AMR. We've got limited antibiotic targets. We've got multiple resistance mechanisms, many of which are transferable because there are mobile genetic elements. We have a discovery void and an empty antibiotic pipeline due to an inappropriate economic model of producing antibiotics. There's no financial return on them, so there's no marketplace, and therefore pharmaceutical companies have left the market. We have a deep-seated societal reliance on antimicrobials, which started way before we used them at sort of industrial scales. And therefore, we keep on using the same antibiotics for the same diseases, and this inevitably increases the selective pressure, which gives rise to increasing levels of resistance, with no real solution on the horizon. So let's turn to the second part of the talk and think about the data insights and the translational value of using whole genome sequencing surveillance. So if we think first of all about the microbiology base, the antimicrobial sensitivity, the AST surveillance, we simply know what bacteria do. We know if they can or cannot grow in the presence of an antibiotic. However, if we incorporate whole genome sequence base, so this is genotypic surveillance, we can get a vast amount of additional information. We can understand the genetic basis for antimicrobial resistance. We can understand whether that's by mutation or by the acquisition of one or more genes, which usually gives rise to resistance phenotypes. Importantly, we can understand the mobile genetic elements responsible for carrying these AMR genes. So this is a particularly useful example of this is with extended spectrum beta-lactamases and their transfer between the Enterobacteriaceae, between E. coli and Klebsiellas. And importantly, we can also understand the evolution of resistance genes, mobile genetic elements, and the bacterial pathogens themselves. We can understand how they are changing in response to different antibiotic regimens in different hospitals in different countries. So this gives us a very nice overview and allows us to think about population dynamics of bacterial pathogens over space and time. So we can draw that spatial and temporal analysis from the various data sets providing the samples exist. I said I was going to mention whole genome sequencing and GLASS, and this is kind of the front cover of it. So the Global Antimicrobial Resistance and Use Surveillance System, also known as GLASS, is a global data repository, and it's aspirational. It is something that many countries aspire to input their data into, as detailed in their national action plans on AMR. And GLASS itself is part of the Global Action Plan on AMR. And it was sort of ratified in October 2015, so it's been going for 6 years, and it was the first global collaborative effort to standardize AMR surveillance. And the important thing about this is that the antimicrobial susceptibility, so the phenotypic data, which is incorporated into GLASS, is standardized. So for the first time, you can have comparison between different countries in different parts of the world in terms of the numbers of different pathogens that are being reported. It's worth saying here that GLASS doesn't incorporate whole genome sequencing data strictly across the board. It's something which it's trying to do, and it does provide a huge amount of advice and recommendations to health systems which are trying to incorporate whole genome sequencing into their surveillance efforts. If we went onto the GLASS website, we would see these different boxes here. And the main one that I want to mention is GLASS AMR. So this is where pathogen-specific data is incorporated. And at the moment, the pathogens which are included are Acinetobacter, E. coli and Klebsiella, Neisseria gonorrhea, Salmonella and Shigella species, Staph aureus, and Streptococcus pneumoniae. So if you're going to add this data to GLASS as a country, so at national level, you will have all of this data for all of these pathogens, and then you fit in to a global picture of the burden from this disease. And then we've also got other data sets within GLASS incorporated within antimicrobial usage, which is interesting. But the second one I want to highlight is this One Health one. So this module was launched in 2018, and it is basically the Tricycle project. It is a project concentrating on E. coli and the production of extended spectrum beta-lactamase enzymes. So it identified E. coli, ESBL E. coli, as a common indicator to be detected across human samples, poultry samples, and within water bodies, specifically sewage and market runoff and river sites in urban areas. And it's done this to try and get a One Health approach. Just as a reminder, One Health incorporates animal health, environmental health, and human health, all in the same framework, so you can understand more fully the sort of transmission events which lead to the dispersal of particular resistances, in this case ESBLs. And that leads me on very nicely to this particular paper here, and some of the work that's been carried out by Nelly Puspandari within Indonesia itself. And you have to think about what to sequence if you're going to introduce surveillance, whole genome sequencing in your surveillance. And the reason why I want to highlight this particular paper is that the tricycle implementation has been carried out, and what you have now in Indonesia is a very nice archive of bacterial strains from overlapping sampling points, which we'll see in a minute. And if you were able to go back in Indonesia or in any country to the archive strains, you can provide a nice foundational knowledge of the circulating resistances at a particular time, at a particular location, whether that be district or hospital or actually national, for a particular resistance. And that's really important if you want to begin to monitor resistance development in that particular situation. So within the tricycle implementation project in Indonesia, you've got specific sample sets from hospitals. So if we just think about whole genome sequence surveillance from hospitals, what are the types of questions that you can answer? So you can answer which ESBL genes are circulating within a specific hospital. Are there differences between hospitals? And are the current antibiotics being used appropriate? And this comes, sort of the power of this approach comes into force when you can detect genes which maybe are not leading to the phenotype. So they would be missed upon microbiology-based antimicrobial sensitivity testing alone. Having that genome sequencing data as well enables you to predict that actually with increased use of a particular antibiotic, you may lead to the selection of the resistance, which is already there in the background. Now, if we expand these sample sets within the tricycle project within Indonesia, what you can see is that you've got this spatial and temporal overlap between human, which is shown here in B, the sampling sites in the hospitals, and also within the environment, so within the slaughterhouses and the market runoff areas there, so with the animal, and also D, which is the environmental within the river systems. So when you have the whole genome sequences available from all of the E. coli in these particular sampling sites, you will be able to determine if there are any transmission events, if it is likely that you are getting, for example, from animal to human transmission from the food markets which have been sampled, or if there is acquisition of ESBL producing E. coli into the water systems downstream from hospitals. And what this will enable researchers to do in Indonesia is to identify if there are any obvious intervention points where there's likely to be most impact from a particular intervention, such as hand washing at animal markets. That's just an example. The data is not available yet, and when it is, it'll be really interesting to see what that sort of throws out. Now, I just want to use another example of a more mature collaboration that we've got at LSTM, and that's with the MLW, Malawi Liverpool Wellcome Trust facility in Malawi, which is run in conjunction with the University of Liverpool and also the College of Medicine in Malawi, funded primarily by the Wellcome Trust. And what that partnership has enabled researchers in Malawi to do is to build up quite a large database, if you like, an archive of tens of thousands of routinely collected bacterial isolates from the main hospital within Blantyre in Malawi. And the samples are from a time frame of 1998 to 2016. And this paper, published a few years ago, highlights the antimicrobial sensitivity profiles that we're seeing. So you can see here that we've got increased resistance to all of the different antibiotics which have been investigated. On this map, we've got ceftriaxone, ciprofloxacin, and gentamicin. And there's similarly an increase in all of the different bacteria which encode this particular resistance. But remember, this is only antimicrobial sensitivity testing. But the fact that these strains are sitting in the freezer means that now we can start sequencing them. So the genomic landscape of ESBL resistance in E. coli, for example, has now been started to be elucidated. 100 or so strains were recently sequenced, and this is published data, showing that we do actually in Malawi have a kind of high representation of the sequence type 131. So this is the global epidemic sequence type. This is found pretty much everywhere. It's able to transmit really easily. But what is interesting within the Malawian context is that there are a lot of different sequence types and phyla groups. And this is unusual because it shows that there's a lot of different phylogenetic groups circulating within a Malawian context. And that's something that we didn't know until the genomes were sequenced. And then we can delve even further and we can start to look at the individual resistance genes which are present in all the strains. So this is the phenotypic resistance. We can match that to the presence or absence of the various genes. And importantly, we can match that to the presence or absence of plasmids of different incompatibility types. So we can see, for example, the sequence type 131s have the resistances we would expect, and they are likely to be on the plasmids that we think they are on, based on what else is circulating on a global level. So this is a really nice way to be able to know exactly what's going on within the strains over and above simply having a phenotype of resistance. And that's just E. coli. If we now think of Klebsiella, so some of the Klebsiellas have been sequenced recently again, and similar analysis can be carried out. We can match the phylogenetic groupings with the presence or absence of the different resistance genes, and also match it with the AMR phenotype through the antimicrobial sensitivity testing. And then we can see if the resistance genes present are actually on particular plasmids, and we can see an overrepresentation of these particular plasmids within this cohort. And then what I wanted to highlight here as well is that this can also act as an early warning system, because one of the strains sequenced of these Klebsiella pneumoniae, and there are about 100 again, actually contained a plasmid which had a very high identity to this plasmid here, which contains the NDM1 carbapenemase gene. Now, carbapennamases are not yet used in malaria. They're not available. They're too expensive. But we've shown here that we've actually got the circulation of an NDM1 gene already. So that's something to be aware of if and when carbapenems are actually introduced for therapy. So we can extend that data. So this is Richard Goodman here. He's one of my PhD students. And he's actually looking now at the co-linkage of different resistance genes within all of these genomes. So now they're published, all of the genome data is online and available for subsequent analysis, which is really good, and it's a really strong way to increase the research which occurs within your data set. And this is just some examples of the co-linkage that he's seen. So this is about 70 strains. Within 9 of them, this actual network exists. So we know that these are likely now to be on the same mobile genetic elements, probably plasmids. And then if we sort of drill down and look at the more common networks, which will inevitably be smaller, we can see the co-association of these 3 genes in more than half of the strains. An analysis of the genome shows that these are present on integron structures. So you can continue to analyze the genome sequences really until your heart is content. So you really have to focus on the actual questions that you want to answer. And this will change when you come from the kind of the clinical aspects, am I using the right antibiotic, down to the more academic aspects, you know, what is happening in the strains, how are they evolving in response to different drugs being introduced in this particular context. So finally, let's just think about the roadmap to implementation. So really, you need strong commitment for whole genome sequencing to be incorporated into your analysis. You need assessment of your capability, and you need the technical development to be able to do it. And for all of those things, you need robust business plans and standard operating procedures. And all this gives rise to an element of sustainability. Now, most of this is actually mapped out in this publication, which is quite recent. And it's a really good sort of template to think about if implementing whole genome sequencing at a national level. But it doesn't necessarily have to be national. It could just be citywide, for example. If we think about the technical infrastructure, what you actually need is very good laboratory infrastructure. You need robust bioinformatic analysis, and we'll come back to how or why you do that in a moment. You need to be able to link the data that you've got with phenotypes through antimicrobial susceptibility testing with the whole genome sequencing. And this is why it's important to gather also the metadata of your samples. And then you also need financial accountability, because this costs money. It's not free, it is expensive. And to do it correctly, you need sustainable financial power. And again, this leads to stability. And all of these particular aspects require training, and they also require internal and external oversight and quality assessment. And you need to think about who's going to train the trainers. If you have a pinch point within your facility and there's only one person who knows how to do one aspect, that's not going to work, because inevitably you get staff changes, etc., etc. So you need to be able to build in suitable training mechanisms so you get a sort of vertical flow of information and expertise throughout the lifetime of the sequencing project. And also you need to think, I think, about who guarantees excellence. If you're going to do it, you need to do it well. You need to get the most from it. And as I say, you need both internal and external quality assessment throughout the lifetime of the sequencing. And hopefully that, once implemented, would be continued. And then I just have to highlight.
Wrap up, sorry.
Yes.
May we wrap up? Yes, thank you.
Last but one slide.
Oh, okay, thank you.
So I just highlight the collaboration here between the Ministry of Health in Indonesia and the Fleming Fund with DAI, who are working towards the implementation of this particular aspect of whole genome sequencing in surveillance. And then my final slide is just to make note really of the huge numbers of bioinformatic resources present. This paper in 2019 detailed 47. They're coming out what seems like every week. So you need to be agile. You need to be able to respond to the tools available and choose the correct tool for your question. So to conclude, whole genome sequencing as part of routine microbiological surveillance is able to greatly expand the knowledge of circulating and outbreak AMR bacteria. And not every AMR gene present will be expressed all the time. So genotype-phenotype mismatch will be seen quite often. And the data can be used directly to inform policy decision-making, such as prescribing practice at local and hospital scales. And it can also serve as an early warning system for new outbreaks and should be thought of before introducing a new antibiotic. And then finally, I would say keep all of your samples. Never throw anything away, because you can always go back and sequence them. And then finally, I'll acknowledge these particular funders and institutes, and particularly Nick Feasy, Patrick Bisicha, and Richard Goodman, who we are continuing the work with. And I'll be happy to take any questions. Thank you.
Thank you so much, Dr. Adam Roberts. Thank you, and very interesting. Now we are moving to the using this genome sequencing for AMR. I guess we will have to move on to the question and answers session. If there's time left, I would like to ask Adam later on. Can we have the questions? Do we have Dr. Lin, Dr. Eggink, and Dr. Robert here? And the committee, can you show us the slides of the questions? I think there were some questions. So, from the NIH itself, I think this is for Dr. Lin. We know that Singapore's vaccinations achievement has been high, but lately there has been an increase in COVID-19 cases. How did this happen? Dr. Raymond, are you still in the room? Okay. Is Dr. Raymond in the room? Let me check the participants. Okay, I think he's left the room or maybe disconnected somehow. Okay, Dr. Raymond is not here. Yeah, I think he's a bit away, and then probably the committee will ask him to rejoin. Okay, Ibu Ratna commented in Indonesia, our government protect our country from new mutations with quarantine and PCR tests for foreigners or someone coming from abroad to Indonesia. What will be your opinion that we need whole genome sequencing test for someone who come or enter Indonesia from abroad? Thank you. Maybe if Dr. Lin is not here, Dirk can maybe suitable for you, the question. This is about the method that we do to protect the country is by quarantine. What do you think?
Yeah, and it's an interesting point because, and of course, Indonesia is a different country than the Netherlands. We're only a small country. We have very much open borders. We have a lot of travel in and out of the country. So if I look at our own situation within the Netherlands, for us, it's impossible to get everyone tested and sequenced that enters the country. That's just from a pragmatic point of view, from a practical point of view, it's impossible. So it depends on that. And another thing to consider there, I think, well, it's the number of travel. Is it possible to test everyone and to isolate everyone that is possible and thereby keeping novel variants? Because if you only test a small portion and at the same time you have a lot of travel that you cannot test, then what does it add to have such strict testing for a small subset? So that is something that you need to see per country. And another thing to consider there, and I don't know what the good or right answer is, but it is something to consider, is what are your measures? So what is your follow-up for that novel variant? Because as I mentioned in my talk, in the Netherlands, at least, we do not have different measures for different variants at this point. We have a general policy in the Netherlands, and our government make rules depending on the number of cases and especially the number of hospitalizations, because we want to make sure that our hospitals can handle it. And although it's only very minimal that they can handle it because there's so much pressure on those hospitals, at this point they are still able to handle it. But that doesn't matter whether it's the alpha, the beta, the gamma, or the delta variant. So therefore, for us in our policy in the Netherlands right now, we do not check every sequence that comes into the country. As I said, for us, sequencing is important to know what is circulating and how can that help us in predict the number of cases, rather than having the plan to isolate every specific case, because the incident and number of cases in the Netherlands is too high right now, and it has been too high throughout the whole pandemic. If incident become really low, so if we have only very low number of cases, then this strategy might be more suitable for a country like the Netherlands.
Thank you. So it's also about what is now currently happening. The incidents itself also influence the policy, what to do with the foreigners. Indonesia has more than 200 airports and thousands of seaports. So
yeah, so it's very hard to actually, like New Zealand did, to kind of close the country. Yeah, that's for countries like Indonesia and the Netherlands, that's a lot harder.
Okay, thank you for that. So I think there's a question again to Dirk. Why not? Oh, I think this is a bit relevant to the previous one. Why not every cases we conducted whole genome sequencing? Oh, I think that was already answered. It's about the same. Pak Hamidi sudah tadi hampir mirip dengan yang pertanyaan tadi ya. Dr. Nasrom mungkin bertanya, di Netherlands juga tidak dilakukan semua kasusnya dengan genome sequencing-nya. Oke, ada pertanyaan berikutnya, how to perform tracing and tracking surveillance using whole genome sequence in an area with limited sequence whole genome sequence data. Pak Dr. Givari, ini ditujukan kepada yang siapa ya? Dr. Dirk atau Dr. Adam ya? Okay. Anyone would like to answer that? What will be then? How do you perform?
I can start, and
Yes.
maybe Dr. Roberts can fill on, because if there's limited sequencing capacity that affects both viral surveillance as well as AMR surveillance, of course, and it's very important that we work together. Collaborations, I think, are very important there, and it's important in that situation, if you have limited sequencing capacity or laboratory capacity, is to really think about how much do I need to sequence to get to what I want to know. So what is the rationale behind the sequencing? And then you can decide on what numbers are necessary. And is it possible to ship samples to collaborators in the country? Can we look for international collaborations? Because that's also what, for example, the Netherlands is doing in the Caribbean part of the Netherlands. So there are a couple of islands in the Caribbean that we help sequencing. We are helping Suriname because there's little sequencing capacity. So we are sending people for training. It's not only the RVM, but also Erasmus MC is doing a fantastic job there. And we're receiving samples. So two things are important. Try to find collaborations within your country or internationally. Reach out via WHO, for example. We're always willing to help if possible. And think about the numbers. How much do you want to sequence, for what reason, and how can we establish that? But that's my opinion.
Thank you, Dirk.
Yeah, I'd like to add to that. I couldn't agree more with Dr. Eggink's analysis of that. Collaboration is key. There's two things I would add to that. One is to save your samples. So even if you don't think at the time you have capacity to sequence everything, don't throw them away. They are absolute gold. You can always go back and sequence if the samples are archived. And then it just becomes a bit of a historical study rather than a contemporary study. But the power of that study and that data can be as good, if not even more useful, when you think about it through a historical lens. And the second thing I would add to Dr. Eggink's analysis is think about the Nagoya Protocol. If you are going to search actively for collaborators and you're going to be thinking about shipping strains or DNA or even data for analysis, think about the Nagoya Protocol, because our our experience at Liverpool School of Tropical Medicine has shown that with various partners, it can sometimes take months, if not years, to sort out a suitable framework within the Nagoya Protocol to exchange samples and DNA. Nagoya Protocol was put in place for the right reasons. It is very cumbersome, however, to navigate through. So start thinking about that aspect as early as possible within the remit of international collaborations. But don't throw anything away.
Okay, I think that comes to the kind of the last question. If we have bank of positive samples from 2020 until now, what strategies we can use by doing whole genome sequencing?
Is that question for me or for Adam?
I don't know. It didn't mention to whom it was.
Well, we can maybe answer them both.
Okay.
Shall I start again? Yeah, if it is about SARS-CoV-2 sequencing, like Dr. Roberts is pinpointing, if you haven't sequenced it yet, then your goal behind the sequencing and your rationale changes, because it becomes an historical study. So then it is important to decide on what do I want to know, what do I want to investigate, for what reason, and then you can make that selection. And sometimes it takes time, because now if we're talking about SARS-CoV-2, we're in the middle of a pandemic, so it's not always easy to think about the scientific questions behind the topic, because it's only, everything is ad hoc. We need to decide now for the measures of tomorrow, but therefore, we have very limited time to think about what information could be in these historical samples. So there, I would, at this point, storing is key, and I'm glad that point was brought up. And if you cannot store everything because there are too many samples, think about a selection method, a random selection method. At least for SARS-CoV-2, random selection is very important, is more important than investigating clusters in general. So that will be a thing. And at this point, for the SARS-CoV-2 pandemic, I would focus on sequencing novel samples and then store the old ones, because when there's a little bit more time and things are getting a little bit more calm, there's more time to think about what are the scientific questions that still need to be answered, and how can these samples add and help in that.
Thanks.
Yeah,
Adam, would you like to add to that?
Yes, please. I agree with you again. A random collection of samples for resistant bacteria is also really useful. I will give you an example. Point in case is the mobilizable colistin resistance. So resistance to colistin was only found to be present on a plasmid and therefore able to transfer in 2015. It was reported from China. China used a huge amount of colistin, upwards of 20,000 tons per year in their pig production facilities. Now, when this gene was found, it made international headlines around the world. And the first thing everybody did, me included, was go back into your strain archive and see if you've got the gene there. And it was actually shown, and this illustrates the benefit of having these archive samples. It was shown that the MCR1 gene was already present in different countries throughout the world prior to, I think, 2012. So it was already circulating before it was originally found in 2015. And that itself highlights the power of genomic surveillance studies being linked to biological studies for actual phenotypes. So it is really important, as random samples help, but I think in a clinical context as well, it's going to depend on when you think you may be able to sequence them. So if you know there's going to be a 6 to 12 month delay, still keep, if you have the capability, all of the samples which are resistant to the antibiotics you are currently using, because that provides that background genomic information, which you need really to look at a response to any change in the antibiotic use within a hospital or nationwide.
Thank you. I think I as an infectiologist, also having a question throughout this pandemic, that in the early in our early guidelines, there is one of the clinical guideline is to give antibiotics to all the SARS-CoV-2. I wonder, Dr. Robert, whether we see any new cases of AMR due to this pandemic. Maybe overuse of antibiotics or antivirus or things like that.
Yeah, so there's definitely an overuse of some antibiotics in primary care. There are some nice studies coming out now which show this is a fact. In terms of impacting on actual antimicrobial resistance, it's still too early to say.
Okay.
It may take years for any effect on the actual bacteria to start to come through the surveillance systems, which is why it's also really important to keep all of your samples through this particular period of the pandemic. With the change in health-seeking behavior, such as the increase in telemedicine throughout the world, prescriptions have changed. There are a lot more prescriptions being given out through telemedicine. However, there are less people engaging with telemedicine. So a lot of people are just coping with their bacterial infections on their own. So that selective landscape of antibiotics has changed. but we don't know yet whether it will have an effect on the genomes of the circulating bacteria themselves.
Okay, thank you. There's a very last question which is interesting. How can we collaborate with you? Dr. Firman Setiawan is asking probably. Mainly major also genome sequencing for AMR. Thank you. How to collaborate with you? Either with the bunkers or I don't know. Anyone wants to add to that?
I just send an email. It's a collaboration is always based on mutual respect and nice conversations, and we work with people that we like. It's a lot easier to work with people that you like. So just send an email and start the discussion.
Okay.
Yeah, fully agree. Not like also for AMR, but also for other things. If you have questions or are looking for collaborations, either for lab capacity or training or doing research together, surveillance together, reach out, send an email. It always starts with that, and from there you see what you can do together. Or if we can't do it, maybe we can help you getting in touch with other parties that you might not know yet, but I might know. So if I can help bringing the right people in touch with each other, so reaching out, that's the first step. Take that step, even if you don't know people, reach out, explain what you're looking for, and then a lot of people around the world are very much willing to collaborate and help.
Okay.
It might be worth just mentioning that if we get 400 emails after this talk, it may take a little while to go through them.
Okay. Yeah, 435 in the room. So I guess we should wrap up. So if you want to walk fast, if you want to go fast, you walk alone. But if you want to go far and sustain, then we collaborate. I think that's kind of the Chinese proverb. And I think that wraps up our session. Thank you so much, Dirk. Thank you so much, Adam, for the very, very interesting talk that you give. And I think it was inspiring, open-minded, and very opening our mind as well. So you are very open-minded to about your knowledge and sharing it with us. Again, thank you, and thank you to all the speakers. And I just have to hand over to the committee. Thank you again,
My
Dirk.
pleasure.
Okay,
My
I think you
pleasure.
can go
Thank
to
you very
sleep
much.
again. Maybe we have another 1 or 2 hours before the sunrise. Okay.
I'm gonna take a little nap. Thank you very much.
Bye.
Bye-bye.
Thank you to our respected speaker and moderator for your availability and time investment to attend our webinar, especially because you have to join this webinar in the early morning. We might disturb your sleep time, but we highly appreciate this. Thank you again. Thank you, Dr. Yanri, for the dynamic discussions and moderation sessions for the first sessions of this webinar. Now, honored attendances, ladies and gentlemen, we jump into the second sessions. For these sessions will be moderated by Dr. Rita Kusriastuti, Master of Sciences. She is an expert for neglected tropical diseases since 2016. She is also a chairman of Indonesian Parasitic Disease Control Association, a vice chair for Global Fund ATM, and other respected membership in different organizations. Ibu Rita, can you hear me?
Yeah.
Okay.
Yeah,
Okay.
clear.
Okay, thank you. Ibu Rita, we will pass the session to you and the floor is yours. Thank you, Ibu.
Thank you. Ya, terima kasih Mbak Uli. Thank you for the beautiful MC. Of course, I will say thank you to Pak Dr. Nana Mulyana and all the Litbangkes team. So now is the turns for Indonesian speakers, dan mungkin saya bisa berbahasa Indonesia, ya, dan juga kemudian karena para peserta semua juga orang Indonesia, ya. So thank you so much. It's an honor of me to be chosen as a moderator in this meeting, ya. Dan for those speakers from abroad, so please now listen the Indonesian words or language, and then you can hear for the what is translators there. Langsung saja karena waktu kita sudah agak terlambat. Ya, pembicara pertama adalah Dr. Ristiyanto. Adakah ya, Dr. Andes Ristiyanto M.Kes. Dia bekerja dari tahun 89 sampai sehari ini sebagai peneliti di Institute for Vector and Reservoir Control Research and Development ya, B2 P2 VRP di Salatiga ya, dan sampai 2019 sampai sekarang juga sebagai kepala of Research Facility and Services ya di Salatiga. Dan beliau menyelesaikan pendidikan doctoral degree di Tropical Medicine Gajah Mada University Yogyakarta pada tahun 2015. Sama ya Pak, kita beda seangkatan nih. Saya juga lulus 2015. Baik, beberapa penelitian begitu banyak, saya tidak akan membacakan. Tinggal cari saja nama Ristiyanto ya dan apa yang diinginkan. Baik, Pak Ristiyanto akan menyampaikan topik the applications of leptospirosis surveillance and its risk factors in public health centers or puskesmas ya, penggunaan aplikasi surveillance leptospirosis di puskesmas. Silakan Pak Ristiyanto, punya waktu sebetulnya 30 menit, tapi kalau bisa sedikit saving 5 menit kita bisa pakai buat diskusi. Oke, please, Pak Ristiyanto, time is yours.
Terima kasih Ibu Rita. Assalamualaikum warahmatullahi wabarakatuh kepada Bapak Ibu sekalian. Salam sejahtera bagi semua. Pada kesempatan ini, pada webinar internasional ini, saya akan membawakan secara bahasa Indonesia dan mengenai penelitian yang kami lakukan selama 2 tahun di Jawa Tengah. Saya akan share screen. Apakah sudah jelas Bapak Ibu untuk PowerPoint yang...
Ya, bisa dilihat.
Terima kasih.
Ya.
Bapak Ibu sekalian, untuk peserta internasional webinar, untuk kali ini tadi saya sampaikan akan menyampaikan tentang aplikasi surveillance leptospirosis dan faktor risikonya di puskesmas. Kira-kira outline dari paparan saya yang jelas mengenai hasil dari penelitian tersebut, yaitu berkenaan dengan pelatihan untuk persiapan surveillance leptospirosis, kemudian penemuan kasus demam akut, kemudian kelompok umur dan pekerjaan, gejala kasus leptospirosis di area penelitian, hasil diagnosis ya yang menyangkut suspek probabilitas, kemudian fluktuasi dari kasus itu sendiri, kemudian surveillance pada hewan ternak, surveillance pada rodent, kemudian epidemiological investigation atau penyelidikan epidemiologi, yang terakhir adalah leptospirosis surveillance pada faktor abiotik. Bapak Ibu sekalian, kita tahu bahwa leptospirosis merupakan zoonosis tersebar di dunia, dan bahkan di Indonesia sebetulnya sebarannya cukup luas dan umum. Menurut Rikus atau riset vektor dan reservoir penyakit di Indonesia, hampir semua binatang liar, khususnya tikus, itu sudah terdeteksi positif leptospirosis di 29 provinsi. Hal ini menunjukkan bahwa faktor risiko leptospirosis itu berada di sekitar masyarakat kita. Sedangkan kejadian luar biasa atau KLB itu sering terjadi di daerah-daerah perkotaan maupun pedesaan, seperti di Jakarta, di Yogyakarta, di Jawa Tengah itu sendiri. Kemudian terakhir ini kami mendapatkan informasi dari Kalimantan utara, Sulawesi. Ini mulai ditemukan leptospirosis yang menyebabkan ratusan orang terinfeksi dan 10%-nya itu meninggal dunia. Jadi leptospirosis ini cukup meresahkan karena di samping dapat menyebabkan kematian, menyebabkan leptospirosis berat dan secara ekonomi jelas akan merugikan masyarakat yang terkena leptospirosis itu sendiri. Yang penting lagi Bapak Ibu, bahwa penyebaran leptospirosis dan peningkatan kasus leptospirosis di Indonesia itu masih belum diketahui. Karena kita tahu baik sistem surveillance-nya, prasarana dan sarana pengendaliannya, kemudian tata laksana kasus, kemudian juga alat-alat untuk mendeteksi kasus leptospirosis yang sangat hampir sama dengan penyakit-penyakit lainnya, itu masih sangat terbatas. Dan yang lebih penting lagi, leptospirosis ini menginfeksi semua binatang mamalia yang ada di sekitar manusia itu sendiri atau di sekitar lingkungan kita. Seperti hewan-hewan liar, terutama tikus, kemudian kucing, anjing, bahkan ternak besar seperti sapi, domba, dan lain-lain. Dan kita tahu bahwa untuk siklus penularannya, leptospirosis yang paling banyak itu adalah melalui lingkungan air yang terkontaminasi oleh bakteri leptospirosis. Dan saat ini di Indonesia leptospirosis hanya banyak ditemukan di rumah sakit yang biasanya sudah dalam keadaan kronis atau boleh katakan merupakan leptospirosis berat atau well disease dan kematiannya cukup tinggi. Nah, kalau di rumah sakit surveillance-nya itu dilakukan secara intermittent dan episodik, sehingga leptospirosis sangat-sangat berbahaya bagi masyarakat yang terkena dan karena menimbulkan kematian cukup tinggi. Oleh karena itu, dalam penelitian ini menerapkan sistem surveillance yang dilakukan oleh puskesmas. Untuk penelitian ini dilakukan di dua daerah, yaitu di daerah Banyumas dan Demak, dan dilakukan pada Januari 2018 sampai Desember 2019. Kemudian kegiatan utama dalam surveillance ini adalah secara pasif maupun secara aktif. Secara kontinu dilakukan di 39 puskesmas di Banyumas dan 27 puskesmas di Demak. Sebelum melakukan surveillance ini, kami melakukan ceramah klinis atau training ke tenaga medis, khususnya dokter, surveillance officer atau juga tenaga laboratorium, terutama mengenai diagnosis, deteksi, dan epidemiologi leptospirosis. Penemuan kasus demam akut, ya kalau kita lihat dari prosedurnya, penemuan kasus demam akut yang memenuhi kriteria secara pasif dan aktif tadi didiagnosis oleh dokter puskesmas. Kemudian diambil spesimen darahnya, selanjutnya dilakukan penyelidikan epidemiologi, survei roden, survei hewan ternak, dan survei lingkungan. Sampel darah itu diperiksa dengan RDT, Rapid Diagnosis Test, di puskesmas, dan diperiksa MRT, Microagglutination Test, dan PCR di B2-P2-PRP. Nah, ini skema dari pengambilan sampel atau spesimen sampel. Jadi dari daerah penelitian kami mengambil darah manusia, hewan ternak, dan tikus, kemudian juga ginjal tikus, serta air dan tanah. Sampel darah manusia diperiksa RDT dan MAT, dan juga PCR. Kemudian darah hewan, baik hewan ternak maupun tikus, diperiksa dengan MAT dan PCR. Sedangkan ginjalnya hanya diperiksa dengan PCR sama dengan air dan tanah. Hasil pemeriksaan tersebut kemudian kita analisis, kita kelola sehingga bisa diketahui angka kesakitan, kemudian seroviral leptospiranya, dan faktor risiko yang berpotensi dalam penularan leptospirosis. Kemudian untuk hasilnya, Bapak Ibu sekalian, bahwa pada pelatihan atau ceramah klinis pada dokter, tenaga surveillance, dan tenaga laboran di puskesmas, itu menunjukkan bahwa ada suatu peningkatan pengetahuan, baik cara diagnosis maupun epidemiologi leptospirosis itu sendiri, baik pada dokter maupun pada surveillance dan tenaga laborannya. Kemudian kita melakukan case finding, yaitu penemuan kasus. Ada penemuan kasus demam akut secara aktif, itu sebenarnya dilakukan oleh kader kesehatan. Kemudian dirujuk ke puskesmas dan didiagnosis oleh dokter puskesmas. Nah, kader puskesmas itu biasanya ada komunikasi dengan masyarakat setempat secara non formal sebenarnya, karena masyarakat di sini belum pernah kami latih untuk bagaimana mengidentifikasi atau mengetahui panas akut yang mengarah ke leptospirosis. Jadi kemungkinan ini secara non formal kader kesehatan menginformasikan ke masyarakat, dan hasilnya beberapa masyarakat ya, itu cukup merespon, jadi berperan aktif melaporkan kasus demam yang ada di sekitarnya, ya terutama di keluarganya. Bahkan tetangganya itu ikut melaporkan ke kader kesehatan. Nah, dari hasil tersebut, karena boleh katakan masyarakat maupun kader kesehatan secara langsung belum dilatih tentang bagaimana menemukan kasus demam akut yang mengarah ke leptospirosis, maka temuan kasus demam itu lebih banyak ditemukan secara pasif, yaitu di puskesmas, daripada secara aktif. Kalau kita lihat lebih jauh lagi, bahwa pada umumnya kasus demam akut itu terjadi yang mengarah ke leptospirosis itu pada umur 18 sampai 55 tahun, dan pada umumnya sebagai pekerja petani atau buruh tani. Sedangkan gejala, nah ini yang menjadi suatu kunci dari surveillance ini, yaitu menggunakan buku pedoman yang diterbitkan oleh Kementerian Kesehatan ya, tahun 2017, yang juga buku itu berdasarkan dari pertemuan WHO dan CRO tahun 2009, di mana salah satu pakar leptospirosis di Indonesia, yaitu Prof. mengikuti pertemuan tersebut dan menuliskan di dalam buku pedoman teknis penemuan kasus leptospirosis di Indonesia. Nah, dalam pedoman teknis tersebut dicantumkan bagaimana menemukan kasus suspek, kasus probable, dan confirm gitu. Nah, memang sebetulnya ada cara mendiagnosis lainnya sebelum pertemuan WHO CRO 2009 di Cenai ini, yaitu menggunakan metode kriteria FIN, tetapi kriteria FIN saat ini memang masih digunakan di beberapa rumah sakit. Yang saya ketahui adalah salah satunya adalah Rumah Sakit Sarjito. Nah, tetapi kalau diterapkan di puskesmas, ternyata banyak yang lebih menyukai diagnosis dengan menggunakan WHO CRO 2009 yang tercantum di dalam buku teknis pedoman penanggulangan leptospirosis yang diterbitkan oleh M.Kes. Dari penegakan diagnosis dengan buku tersebut oleh dokter puskesmas itu menunjukkan bahwa dokter puskesmas itu telah bisa mendeteksi leptospirosis suspek, probable, bahkan juga terkonfirmasi. Nah, di dalam penelitian ini kami memang menggunakan semua metode baku dalam pemeriksaan leptospirosis, yaitu baik itu dengan MAT, PCR yang tadi kami sampaikan untuk mendeteksi kasus demam akut yang disaring oleh dokter puskesmas dengan menggunakan buku pedoman penanggulangan leptospirosis yang diterbitkan oleh Kementerian Kesehatan. Pada surveillance ini memang ditemukan confirm, bahkan ditemukan juga probable, tapi tidak ditemukan kematian. Karena cepat penanganannya, yaitu cepat diobati, cepat dirawat, sehingga kematian di dalam surveillance leptospirosis di puskesmas itu tidak ditemukan kematian. Yang penting lagi, kita bandingkan dengan kasus berat leptospirosis atau well disease leptospirosis yang ada di rumah sakit dan kematian. Dari hasil surveillance leptospirosis di puskesmas itu mempunyai dampak dan harapannya memang harusnya berdampak terhadap kasus berat maupun kematian yang ada di puskesmas. Dampak tersebut terlihat bahwa dengan adanya surveillance kasus leptospirosis di puskesmas ternyata bisa menurunkan demam atau boleh dikatakan leptospirosis berat, well disease, sebesar 33,24%. Sedangkan kematian itu cukup bermakna juga, yaitu 24,31%. Walaupun sampai saat ini masih tetap ada kematian-kematian leptospirosis, tapi bisa ditekan serendah mungkin. Kemungkinan kalau surveillance ini dilakukan secara kontinu, terus-menerus, kemudian bisa ditekan lebih besar lagi untuk penurunan kasus kematian leptospirosis atau leptospirosis berat. Bagaimana mengenai surveillance rodent untuk leptospirosis ini? Kita lihat bahwa kita juga melakukan surveillance rodent secara intensif setiap bulan bahkan di daerah di mana kasus leptospirosis itu ditemukan. Ternyata terlihat bahwa tikus yang positif itu selalu menyertai atau selalu ada di daerah di mana kasus leptospirosis itu ditemukan. Dan setelah kami hitung rasionya, ternyata antara kasus leptospirosis dan tikus infektif itu antara 1:3. Sedangkan rasio tikus positif dan tikus yang ditangkap atau jumlah tikus tertangkap itu 1:12. Nah, kita tahu bahwa surveillance tikus secara rutin di daerah endemis leptospirosis itu membutuhkan anggaran, tenaga, dan waktu yang relatif banyak. Oleh karena itu, dengan diketahui rasio antara kasus leptospirosis, tikus infektif, dan jumlah tikus tertangkap, maka surveillance tikus itu dapat hanya dilakukan secara spot survey saja, ya, tidak dilakukan secara kontinu seperti itu, sesuai dengan keperluan. Ya, misalnya saat terjadi KLB atau setahun sekali misalnya, tanpa pemeriksaan laboratorium atau MRT atau PCR, itu khusus untuk tikus. Nah, hasilnya kita hanya bisa melihat rasionya saja. Dengan melihat rasionya tersebut, kita bisa langsung melakukan suatu pengendalian tikus. Jadi lebih efisien. Jadi masyarakat sebagai ujung tombak dalam melakukan pencegahan penularan leptospirosis. Kemudian yang penting lagi adalah hewan ternak. Di sini di dalam survei leptospirosis pada hewan ternak, kami menemukan sekitar 11,53%. Jadi hewan ternak itu sebetulnya juga cukup berpotensi sebagai sumber penular leptospirosis di lingkungannya. Nah, disinilah pentingnya One Health atau kerjasama antar sektor, terutama untuk dinas pertanian dalam mengelola ternak agar bebas dari leptospirosis. Sedangkan dari penyelidikan epidemiologi atau epidemiologi investigation, kita menemukan bahwa hampir semua, ini rata-rata, kalau kita rata-rata, itu 77,24% air yang ada di rumah kasus atau air yang digunakan sehari-hari, apalagi yang di sekitar rumahnya, itu tidak terdesinfektan atau tidak menggunakan desinfektan yang terstandar seperti itu, ya. Kemudian di samping itu untuk sanitasinya juga masih kurang. Kemudian penduduk saat melakukan pekerjaan yang berhubungan dengan air kotor, seperti setelah membersihkan selokan, kemudian membersihkan air kubangan karena hujan, seperti itu tanpa perlindungan. Nah, itu kemungkinan menjadi faktor risiko yang penting di dalam penularan leptospirosis di daerahnya. Bagaimana mengenai lingkungan? Untuk lingkungan, kita lihat bahwa perubahan faktor abiotik seperti pH air, kemudian temperatur udara, bahkan kelembapan, itu hampir-hampir sedikit sekali perubahannya. Jadi fluktuasinya itu sangat kecil sekali. Nah, ini pengamatan kami selama 2 tahun. Jadi hampir-hampir sama, ya, selama 2 tahun itu. Oleh karena itu untuk pengendalian leptospirosis, pengelolaan lingkungan itu lebih dikedepankan sebaiknya, ya, seperti meningkatkan pemberdayaan masyarakat dalam melakukan PHBS atau perilaku hidup bersih di lingkungannya, daripada melakukan pengawasan faktor abiotik tersebut secara rutin seperti itu. Itu mungkin kurang efisien. Jadi lebih baik mengedepankan PHBS di lingkungannya. Kira-kira seperti itu. Dari penelitian tersebut, maka disimpulkan bahwa kriteria diagnosis leptospirosis pada buku petunjuk teknis pengendalian leptospirosis yang diterbitkan oleh M.Kes itu dapat diterapkan, ya, dengan baik dan mudah untuk mendeteksi atau mendiagnosis semua kasus leptospirosis yang ada di puskesmas, terutama yang suspek dan probable. Karena pada diagnosis dini atau pengobatan cepat untuk kasus-kasus leptospirosis itu dapat mengurangi tingkat kematian, ya, harapannya dapat mengurangi tingkat kematian, bahkan juga mengurangi kasus berat di rumah sakit atau bahkan juga kematian di rumah sakit. Gitu. Kemudian tikus yang positif, kita tahu tadi tikus yang positif terinfeksi bakteri leptospirosis itu selalu ditemukan bersama kasus leptospirosis di lingkungannya. Ini merupakan hal yang perlu mendapatkan perhatian, terutama untuk penduduk, agar bisa mencegah leptospirosis secara mandiri. Kemudian ternak itu merupakan salah satu sumber juga untuk penularan leptospirosis, karena banyak serovar pada ternak, pada tikus itu juga sama dengan serovar yang ditemukan pada manusia. Kemudian sanitasi yang buruk, desinfektan yang tidak digunakan dalam pengelolaan air, kurangnya perlindungan secara individu, yaitu tidak menggunakan APD di dalam bekerja di lingkungan air yang terkontaminasi, itu merupakan faktor risiko yang cukup bermakna di dalam kejadian leptospirosis di daerah endemis leptospirosis di daerah penelitian tersebut. Sedangkan rekomendasi yang muncul pada penelitian ini, ya, jelas pedoman pengendalian leptospirosis di Kementerian Kesehatan itu bisa diterapkan di semua puskesmas, ya, untuk memperkuat diagnosis dini dan pengobatan leptospirosis secara cepat dan tepat. Kemudian rasio kasus leptospirosis dan tikus positif berpotensi digunakan sebagai sistem peringatan dini dan upaya pengendalian leptospirosis. Kampanye atau penyuluhan secara intensif tentang perilaku hidup bersih dan sehat kepada masyarakat itu perlu dilakukan. Kemudian kerjasama lintas sektoral untuk mensukseskan One Health itu perlu diintensifkan dan diperluas bahkan seperti itu. Kemudian sumber air dan air kebutuhan sehari-hari, nah ini penting sekali di masyarakat. Nah, itu perlu adanya suatu penggunaan desinfektan. Kalau biasanya di PDAM itu adalah kaporit, ya, sesuai dengan peraturan yang ada. Itu kira-kira hasil dari aplikasi surveillance leptospirosis di daerah endemis leptospirosis Demak dan Banyumas di Jawa Tengah. Semoga informasi ini dapat bermanfaat bagi kita semua. Terima kasih atas kesempatannya untuk menyampaikan hasil penelitian ini dan harap bisa digunakan sebagai kebijakan pengendalian leptospirosis di Indonesia. Terima kasih.
Terima kasih. Ya. Terima kasih Dr. Ristiyanto, penelitiannya sangat menarik ya. Tikus ada di mana-mana, leptospirosis berarti ada di mana-mana karena rasionya 1:3 tadi. PHBS, terima kasih pada COVID nih kita sekarang cuci tangan menjadi kebiasaan nih. Cuci kaki juga. Kemudian kerjasama dengan One Health dan tadi sumber air di masyarakat. Terima kasih banyak Pak Riz, mudah-mudahan ada pertanyaan dari peserta, silahkan di Q&A untuk dilihat, dan peserta yang mau bertanya silahkan. Nah sekarang selanjutnya pembicara yang kedua. Pembicara yang kedua yang sangat-sangat menarik juga adalah Profesor Dr. Tjandra Yoga Aditama. Mana slide-nya? Sebentar, saya lihat sebentar. Speaker Gallery. Oke, Profesor Dr. Tjandra Yoga Aditama, spesialis paru konsulen, Mars, DTMH, DTCE, FISR. Wah, singkatannya apa tuh, Prof? Nanti panjang betul. Beliau saat ini adalah direktur dari postgraduate, berarti S2 ya, Prof, IHRD University. Beliau adalah profesor di bidang paru-paru, pulmonology and respiratory medicine di Universitas Indonesia. Juga adjunct profesor di Center for Environment and Population Health di Griffith University Australia. Masih ngajar ya, Prof. Aktivitas lain adalah sebagai board member dari CHEA Med, juga member dari COVAX, yaitu suatu independent allocations of vaccine group. IAVG, dan juga anggota dari expert team dari Presidential Advisory Council. Beberapa posisi 2018 sampai 2020 pernah menjadi direktur untuk infectious diseases di WHO Searo di India ya, Prof. Kemudian 2014-2015 kepala dari Badan Litbangkes Ministry of Health Indonesia. Dan 2009-2014 adalah bos saya, dirjen dari P2 itu masih P2 ya, bukan tidak ada PL-nya, sudah tidak ada PL. Oh, masih ada P2 PL daripada Ministry of Health. Di situ saya bersama-sama, saya stafnya Pak Tjandra. Jadi senang sekali pada hari ini bisa bersama-sama Prof lagi. Dan Prof akan menyampaikan topik adalah tentang menuju eliminasi TB di tahun 2030. Prof, waktu kami persilahkan 30 menit Prof tersedia, tapi kalau bisa menyediakan 5 menit buat questions and answers time. Terima kasih. Silahkan Prof.
Ya, makasih Bu Rita. Saya share screen-nya, mudah-mudahan kelihatan sebentar. Ini selalu slideshow yang pertama lama, saya nggak tahu kenapa.
Share.
Jadi memang judulnya eliminasi tuberkulosis, tapi karena tema kita hari ini tentang surveillance berbasis lab, maka saya akan akan bicara tentang surveillance berbasis lab untuk tuberkulosis. Dan karena ada permintaan untuk whole genome sequencing seperti tadi 3 pembicara yang awal, tentu saya akan isi dengan whole genome sequencing buat tuberkulosis. Kenapa saya nggak ambil whole genome sequencing buat tuberkulosis sesuai dengan permintaan Bu Cahaya kepada saya, karena tidak terlalu mudah untuk menceritakan whole genome sequencing untuk tuberkulosis apalagi di Indonesia. Karena itu saya mulai dengan yang lebih makro saja. Eliminasi tuberkulosis seperti kita ketahui sudah dicanangkan oleh Presiden pada awal tahun 2020, sementara kita tahu saat ini posisi Indonesia seperti ini ya. Ini kalau melihat Global TB Report 2021. Jadi tentu teman-teman sudah tahu yang kotak merah ini, yang hurufnya seperti huruf T, adalah negara dengan high burden TB. Ada Indonesia di sini. Yang biru ini, yang bentuknya seperti apa? Ini seperti begini, itulah negara yang high burden MDR RRTB. Ada Indonesia juga di sini. Sementara yang huruf yang warna kuning seperti ini, seperti apa ini nggak tahu begitu. Yang kotak yang ini, itu adalah negara dengan high burden TB HIV, di mana Indonesia juga ada di sini. Jadi negara-negara ini yang tertulis di sini di tengah-tengah ini, mereka adalah high burden untuk TB secara keseluruhan, high burden untuk MDR dan RRTB, dan high burden juga untuk TB HIV, dan Indonesia dari sini. Saya selalu sampaikan bahwa tadinya tetangga kita Thailand itu juga high burden buat MDR TB. Baru bulan Maret atau bulan Mei 2021 sambil menangani COVID, mereka bisa keluar dari MDR high burden MDR TB ini. Jadi mudah-mudahan Indonesia bisa keluar, kalau toh tidak dari tiga-tiganya, paling nggak keluar satu dulu gitu, baik TB HIV atau MDR RTB, atau kalau bisa keluar dari tiga-tiganya. Ini gambaran sesuai dengan Global TB Report 2021. Kemudian kita sudah, kalau bicara orang bolak-balik ngomong ranking 1, ranking 2 segala macam begitu ya, mungkin tidak terlalu baik, tidak terlalu pas juga untuk dibicarakan, tapi anyway kalau Global TB Report 2021 sih angkanya seperti ini. Nomor 3 terbanyak di Indonesia, India 26%, China 8,5, dan Indonesia terus 8,4 gitu ya. Jadi seakan-akan lebih baik karena Global TB Report 2020, Indonesianya nomor 2, Cina-nya nomor 3, tapi kita lihat angkanya cuma beda dikit-dikit aja begitu ya. Jadi saya kira ini cuma ya, apakah karena Cina-nya sedikit lebih jelek atau karena ada masalah perhitungan, saya tidak tahu. Tapi anyway, kalau mau bicara angka resmi per Global TB Report, maka Indonesia TB-nya nomor 3 di dunia, bukan nomor 2 lagi. Nah, Global TB Report 2021 ini berbeda dengan Global TB Report setiap tahunnya. Saya kira yang biasa menangani TB selalu mengikuti buku Global TB Report, karena Global TB Report 2021 ini secara spesifik bercerita tentang apa dampak COVID terhadap program tuberkulosis. Analisanya ada banyak, tapi salah satu yang paling besar disebut adalah ada penurunan dari TB notification. Ada shortfall dari TB notification pada tahun 2020 dibandingkan dengan tahun 2019 karena tentu saja karena COVID. Jadi the most obvious impact adalah drop in number of people newly diagnosed with TB and reported notifikasi turun. Dan ada 16 negara yang menyumbangkan paling banyak notifikasi di dunia begitu ya, bukan gara-gara lah. Jadi 93% dari penurunan itu terjadi akibat penurunan di 16 negara ini. Dan ya kita mesti akui bahwa Indonesia nomor 2 ya. India memang nomor 1 banyak banget begitu, tapi Indonesia nomor 2. Jadi ini menunjukkan betapa besarnya dampak COVID terhadap penurunan notifikasi kasus. Nah, kalau kasusnya tidak ditemukan, itu tidak bisa diobati dan kemungkinan terjadi penularan di selanjutnya begitu ya. Ini beberapa data saja yang tidak juga terlalu menarik buat kita begitu ya. Ini largest relative reduction annual notification. Ada Gabon, Filipina, ini negara mana nih? Gabon, Filipina, Lesotho begitu, Indonesia. Jadi temennya kok nggak terlalu bagus-bagus gitu ya. Ini global drop TB case notification. Ada Angola, ada Indonesia, ada Uganda, ada Ukraine dan seterusnya. Kalau ini gap antara MDR yang diperkirakan ada dengan MDR yang berhasil diobati, nanti akan kita bahas juga. Ini juga sama, Kongo, India, Indonesia lagi, begitu, Filipina juga. Jadi memang artinya hanya ingin saya sampaikan bahwa situasi kita memang masih cukup berat, apalagi gara-gara COVID-19 itu. Tetapi kan memang targetnya eliminasi, dan target eliminasi ini sangat ambisius begitu, cukup kuat begitu, cukup besar begitu ya. Jadi penurunan insidence itu 5 kali lipat dari 312 per 100.000, targetnya 2030 menjadi 65. Dan 2030 itu nggak jauh ya, sekarang sudah 2021, hampir 2022, jadi mungkin 8 tahun setengah lagi harus turun 5 kali lipat gitu. Apakah mudah-mudahan bisa, tentu saja. Dan kematian juga tadinya 34 per 1.000 menjadi 6, jadi turunnya 5 kali lipat gitu ya. Memang kita punya perpres, dan ini seingat saya hanya sedikit atau bahkan nggak ada penyakit yang ada perpresnya secara spesifik gitu. Kalau ada mungkin teman-teman bisa sampaikan, tapi perpres ini jelas merupakan modal dasar gitu ya. Sehingga mudah-mudahan sih Ini saya nulis ini judul ini bukan dari saya. Ini korannya yang membuat judulnya. Korannya membuat judul secara apa namanya? Bye-bye epidemi tuberkulosis begitu 2030 yang kemudian banyak orang yang bilang itu judul terlalu keras, tapi anyway itu judul memang dari korannya dan mereka punya teknik sendiri untuk membuat judul seperti itu. Nah, sekarang kita bicara untuk bisa menurunkan ini tentu harus ada kegiatan. Nah, berhubungan dengan judul kita tema surveillance berbasis lab, maka semua bukan semua, kita tahu bahwa diagnosis tuberkulosis saat ini sesuai dengan surat edaran Dirjen, saya nggak tahu ini SK M.Kes atau SK Dirjen, yang mengatakan semua yang terduga TB harus diperiksa tes cepat molekuler. Ya, kalau positif, dan ini ada urutannya, kalau dia negatif ini yang dilakukan, kalau hasilnya error seperti ini, kalau TCM-nya positif resisten rifampicin seperti ini, kalau positif tapi rifampicin-nya masih sensitif seperti ini, kalau intermediate diulang lagi gitu ya. Dan ini teman-teman yang bermain di bidang tuberkulosis ini dibahas cukup panjang algoritma ini, tapi pokoknya menunjukkan bahwa kalau kita bicara surveillance berbasis lab, basisnya sekarang diagnosis adalah tes cepat molekuler. Nah, orang kan tanya ini gimana sih emang bisa begitu ya? Nah, ini datanya ternyata yang sudah ada di Indonesia sekarang tes cepat molekuler ini saya dapat data dari substansi TB per 4 Oktober 2021 ada sekian mesin, sudah ada di 34 provinsi, hampir 500 distrik, sekian pelayanan kesehatan. Jadi sebenarnya penyebarannya cukup banyak ya. Jadi kalau pasiennya memang sesuai dengan gambar yang ini harusnya diperiksa. Kalau nggak ada di dekat dia harus dikirim dahaknya untuk diperiksa tes cepat molekuler sesuai dengan jejaring surveillance lab yang ada di Indonesia untuk tes cepat molekuler. Tentu ada juga jejaring untuk kultur ya. Ini yang jejaring kultur tapi hanya 24 di seluruh Indonesia. Jadi saya kira ini tentu, bukan saya kira, pasti belum ada di semua provinsi, tapi ini ada 24. Saya tanya terus terang saja ke teman-teman di TV. Jadi bagaimana ini ceritanya sekarang, berapa banyak sudah kita berhasil menggunakan tes cepat molekuler yang memang dianjurkan oleh WHO ketimbang menggunakan ketimbang orang yang nggak pakai begituan gitu ya. Nah, ini adalah persentase penggunaan tes cepat molekuler yang memang naik 51% dari waktu ke waktu, naik berdasarkan data ini. Seperti tadi pagi saya sebelum ke acara ini saya telepon dokter Luki yang mengurusi ini di TB. Saya bilang ini apa, ini datanya sudah data nasional semua gitu ya. Tapi ternyata memang ini data yang terlaporkan. Jadi bukan tidak mungkin ada data yang tidak terlaporkan kalau kita mau jujur. Kalau kita bandingkan berapa yang terkonfirmasi bakteriologis di lab dengan hanya terkonfirmasi klinik, ya, jadi tidak ada periksa lab atau tidak ada hasil lab, yang terkonfirmasi bakteriologi naik dari 47 jadi 52, sementara yang terkonfirmasi klinik turun dari 53 menjadi 48. Cuma gambarnya agak tricky ya, ini naiknya kayaknya curam banget, padahal kan cuma dari 47 ke 52, mestinya dibikin lebih landai. Cuma ini memperlihatkan ini naik. Jadi artinya paling tidak dari data yang masuk lebih banyak yang sudah terkonfirmasi bakteriologis, baik DCM maupun juga kultur atau BTA, ketimbang orang yang hanya didiagnosis berdasarkan diperiksa aja atau batuk aja atau ronsen aja begitu ya. Jadi paling tidak situasinya sudah lebih baik untuk menunjukkan peran surveillance berbasis laboratorium. Nah, itu kan untuk diagnosisnya, artinya untuk mengobati. Tapi tentu saja salah satu peran pemeriksaan genome sequencing ini adalah untuk sensitivitas ya, resistensi. Dan di Indonesia ada 14 lab yang bisa memeriksa susceptibilitas, drug susceptibilitas. Ada 14 ini, jadi tidak terlalu banyak memang. Dan apalagi kalau mau pakai teknologi yang baru dengan line probe assay hanya berapa ini? Hanya 7. Gitu. Nah, jadi harusnya seperti kita ketahui, kalau gene expertnya hasil yang menunjukkan rifampicin resisten, maka harusnya ditindaklanjuti dengan CDST, artinya untuk tahu apakah resisten yang lain bagaimana begitu, atau diperiksa dengan line probe assay. Dan datanya di sini hanya 42% dari yang rifampicin resisten menurut gene expert ya, yang terdaftar sekali lagi hanya 42% yang diperiksa kulturnya, resistensi maksud saya, dan 39% diperiksa line probe assay tahun 2020. Angkanya di tahun 2021 karena belum selesai, mungkin tahunnya juga ya 37% versus 39%. Jadi belum semua yang resisten rifampicin di gene expert ditindaklanjuti, dan harusnya tentu semua ditindaklanjuti, dan tentu saja belum semua terperiksa gene expert seperti tadi sudah dibahas. Ya, ini yang hasil kultur ternyata dari tahun 2020 ada 2.700 yang rifampicin resisten, 1.300 yang MDR, pre-XDR 280, dan yang tahun 2021 dilaporkan ada 10 yang XDR. Itu data yang ada, dan ini per provinsi lagi gitu ya, seperti tadi. Jadi 56% yang MDR, dan saya kira 47% yang XDR atau sebaliknya. Pokoknya ini fluktuasinya. Nah, ini hasilnya. Ini bukan surveillance berbasis lab, tapi ini bagaimana performance kita menangani drug resistant. Kenapa drug resistant yang penting? Karena sekali lagi penggunaan gene expert secara lebih detail itu lebih banyak di TB digunakan untuk menangani yang resistensi. Jadi saya ambil tahun terakhir aja deh ya. Dari 24.000 kasus resisten yang diperkirakan di Indonesia, yang ditemukan hanya 5.318, 22%. Dari 5.318 yang ditemukan, yang masuk dalam pengobatan hanya 2.819, 53%. Jadi masih jauh ya, artinya dari 24.000 ini hanya 2.819 yang masuk dalam pengobatan. Nanti kita lihat hasil pengobatannya lebih rendah lagi begitu ya. Jadi memang masih dibutuhkan effort besar untuk menangani drug resistant ini, antara lain dengan menggunakan teknologi molekuler seperti gene expert atau genome sequencing yang kita bicarakan dari pagi tadi. Nah, jadi challenges-nya untuk drug resistant TB di Indonesia, nomor 1, case finding tadi hanya 34%. Yang dari 30% itu yang diobati cuma 57%. Dari 57% yang diobati itu hanya 47% yang sukses rate. Jadi ada 50% lagi yang tidak berhasil diobati, macam-macam loss to follow up dan sebagainya. Dan pada kenyataannya tidak semua 514 kabupaten kota yang punya initiation center ini untuk pengobatan, dan LPA dan CDST kultur tidak tersedia di semua provinsi. Nah, tadi sudah disampaikan bahwa tidak semua pasien yang resisten rifampicin itu diperiksa lagi. LPA dan CDST padahal ini kan harus diperiksa kan untuk tahu resistennya apa saja begitu ya. Dan kita punya gene expert, punya LPA, punya CDST, walaupun jumlahnya terbatas, tapi selain jumlah terbatas juga masih underutilized secara umum. Nah, saya tanya ke teman-teman di sini, gimana rencananya kamu punya sekarang masih terbatas, gimana prospeknya? Nah, ini prospeknya sampai tahun 2024 diharapkan jumlah laboratorium yang naik, jumlah ini uji kepekaan ya, jumlah biakannya naik jadi sekian, jumlah TCM-nya naik jadi sekian, sehingga diharapkan tahun 2024 75% pasien itu terdeteksi dengan TCM, tidak lagi dengan klinis atau dengan BTA segala macam. Jadi angkanya diharapkan 75% sesuai dengan prosedur yang ada, panduan yang paling terbaru. Ini harapan 2024. Tentunya kalau menuju eliminasi TB ini akan meningkat lagi di menjelang 2030, hasilnya akan lebih bagus lagi. Ini targetnya ya, kita ikuti saja tahun-tahun berikutnya. Mudah-mudahan ada webinar lagi, tapi tahun depan nggak mungkin ada webinar dari Litbangkes lagi ya, karena namanya Litbangkesnya udah berubah, jadi webinar dari badan yang baru barangkali. Tentang whole genome sequencing, begitu beberapa informasi yang ada. Pada umumnya sekarang dilakukan pada strain yang tumbuh di kultur. Memang ada yang mencoba pada sputum, tapi hasilnya masih bervariasi, ada yang berhasil bagus, ada yang tidak. Tapi kalau pada strain yang di kultur hasilnya memang cukup bagus. Dan ini tiga kemungkinan penggunaan whole genome sequencing ya. Ini saya diskusi juga sama Bu Vivi, sama Bu Cahaya juga bilang, jadi apa sebenarnya yang bisa dipakai gitu kan? Ya mungkin nanti Pak Nana kalau Mas mengikuti juga bisa, karena ini saya tahu yang ditanyakan oleh pimpinan, bisa nggak dipakai whole genome sequencing yang sudah populer untuk COVID ini dipakai untuk TB juga. Jadi ada 3 saat ini yang mungkin dipakai, walaupun ini perpustakaannya sudah beberapa tahun yang lalu. Pertama untuk, dari tadi saya sebut susceptibility test, yang kedua untuk epidemiological analysis, yang ketiga untuk research. Jadi ini masing-masing achievement dan limitation masing-masing. Gitu ya. Yang paling banyak memang paling banyak memang untuk penggunaan uji kepekaan, ya, uji kepekaan. Dan ini dokumen WHO saya minta dari teman-teman di CRO bagaimana sih pendapat WHO begitu untuk penggunaan genetic sequence untuk surveillance of drug resistant begitu. Tadi sepintas saya dengar tentang AMR, maaf tadi saya kerja yang lain, tapi saya dengar tentang AMR dan ini bagian penting dari AMR tentu saja. Jadi penggunaannya bisa untuk disease burden, untuk monitor time trends, untuk diagnosis dan treatment bukan perorangan begitu ya, tapi lebih ke intervention control, bagaimana effective treatment dan bagaimana policy. Jadi itu kira-kira yang dapat dimanfaatkan dan approach-nya whole genome sequencing. Ini saya terus terang aja nggak begitu ngerti-ngerti banget begitu ya, tapi inilah yang disampaikan teman-teman saya dari Seattle. Dan kemudian kita tahu bahwa bulan Juni 2021 WHO mengeluarkan the first catalog of mutation of Mycobacterium TB dan hubungannya dengan drug resistant. Ini katalog yang pertama baru keluar pada 25 Januari 2021 berdasarkan data dari sekian puluh negara, 40 negara. Kalau saya lihat bukunya, di petanya sih Indonesia nggak ada ya, tapi saya tanya sama Bu Lolong, nampaknya kita sudah berproses untuk mengirimkan, tapi mungkin belum terkirimkan atau belum masuk, sehingga anyway, kita belum masuk rasanya di buku yang Juni 2021, mudah-mudahan bisa masuk di Juni 2000 berikutnya. Pada dasarnya mereka cerita tentang, kita tahu bahwa kalau rifampicin resisten nggak ada masalah lah, itu bisa dianalisis dengan 81 base pair-nya RPOB, udah lama kita tahu ini, tapi untuk yang lain bagaimana gitu ya. Jadi mereka membuat katalog seperti ini, ada 17.000 mutasi dari 38.000 isolate dari 40 negara, seperti saya sampaikan tadi, terhadap 13 antituberkulosis. Selain kalau rifampicin sekali lagi sudah kita ketahui, batuk-batuk itu agak berbahaya, kita lagi ngomong tuberkulosis, jadi Bu Rita hati-hati begitu ya. Ini beberapa obat yang dicek resistensinya, gitu. Tentunya termasuk yang baru-baru delamanid, bedaquiline, dan ini hasilnya yang saya dapat, tapi saya kira sangat teknis sekali, saya juga terus terang nggak ngerti seperti apa, tapi ini hubungannya, ini yang bedaquiline, ini yang clofazimin, ini yang delamanid, dan ini yang linezolid, gitu ya. Ini informasi yang keluar secara detail. Nah, Badan Litbang juga sudah lama melakukan ini, tuberculosis whole genome sequencing study 2020, memang hasilnya belum keluar, makanya namanya preliminary result, dan ini di sini mereka menyebutkan, atau tim menyebutkan bahwa whole genome sequencing ini saat ini adalah salah satu alternatif dalam WHO untuk diagnosis dan deteksi resistensi, gitu ya. Tentu saja juga bisa mendeteksi genetic diversity dan also transmission dynamic. Kalau ada, kita dari dulu sudah dari awal kita pernah dengar strain Beijing dan sebagainya, itu kan antara lain karena itu. Jadi kalau buat multiple drug resistant, whole genome sequencing ini cukup baik, tentu saja lebih cepat daripada phenotypic DST yang bisa makan waktu hari atau bulan. Temuan memang masih susah, masih mahal, butuh laboratorium dan sebagainya begitu. Karena itu dilakukan penelitian oleh Badan M.Kes untuk the whole genome sequencing, karena tadinya saya ingat waktu saya masih di sana memang ada TB drug resistance survey, dan ini kelanjutannya, gitu ya. Kelanjutannya bagaimana dari data drug resistant kemudian didapatkan whole genome sequencing itu, dan saya sudah cek tadi malam lagi, menurut Bu Dina hasilnya belum lengkap, makanya saya nggak berani tampilkan. Ini kalau di slide yang pertama saya kasih ke Bu Cahaya, saya kasih tempat-tempatnya, tapi karena ini belum lengkap, jadi saya kasih gambar umum seperti ini aja. Semua masih under review. Nah, jadi sebagai kesimpulan, secara umum sekarang whole genome sequencing belum digunakan secara klinikal sehari-hari di tuberkulosis tidak seperti katakanlah misalnya COVID begitu. Walaupun COVID juga nggak sehari-hari, tapi kan ada harapan whole genome sequencing ditingkatkan pada COVID, karena jumlah pemeriksaan whole genome sequencing kita untuk COVID dibandingkan dengan Singapura atau dibandingkan dengan Filipina atau dibandingkan dengan India itu masih jauh lebih kecil yang ada di GCA. Jadi kalau kita bilang kita nggak ketemu misalnya ATY 4.2, bisa memang karena tidak ketemu, tapi kalau jumlahnya diperbanyak, ya situasinya bisa berbeda begitu ya. Tentu saja di masa depan mungkin saja whole genome sequencing ini bisa dipakai, terutama kalau harganya lebih murah, dan saya sudah sampaikan sekarang penggunanya lebih ke untuk drug resistant surveillance, dan kalau perlu ada hotspot making gitu ya. Dan ini beberapa pertimbangan practical consideration kalau mau digunakan di masa yang akan datang, memang dia hasilnya lebih cepat, biayanya lebih mahal tentu saja, sensitivity tentu meningkat. Seperti saya sampaikan tadi, kalau nanti bisa lewat sekutum tentu akan lebih bagus lagi, begitu ya. Kalau ada juga cara yang user friendly akan lebih bagus lagi, resolusi lebih bagus untuk analisa epidemiologik dan bisa data, dan ini bisa multi business platform tentu saja ya. Tadi AMR, saya dulu juga mengurusi AMR di CRO, jadi saya tahu gimana proses sampai gelas yang tadi disebut-sebut terjadi dari awal. Dan di masa datang kita berharap ini bukan Indonesia, ini general ya, ada in-country capacity untuk molecular biology and bioinformatics. Jadi saya kira harapannya sesudah webinar ini terjadi, kalau mungkin Pak Nanang dan teman-teman bisa membuat ini terwujud begitu, in-country capacity for molecular biology and bioinformatics. Dan saya baca di media Pak Menteri berulang kali menyampaikan tadinya jumlah laboratorium whole genome sequencing cuma segini, sejak saya masuk naik jadi segini gitu ya, dan itu bisa dipakai mudah-mudahan untuk yang lain. Tentu saja kalau ada yang low cost dan automatic itu akan lebih bagus, lebih mudah dipakai di daerah, ada kit base lebih bagus, kemudian ada tools yang lebih mudah, quality jadi penting, dan kalau mungkin berhubungan dengan surveillance system dan clinical decision. Jadi ini beberapa hal next step yang bisa dilakukan di masa yang akan datang. Jadi kesimpulannya kira-kira begitu. Juga tadi sudah disebut sebenarnya TB is the largest and oldest antimicrobial resistant surveillance project globally. Jadi teman-teman, waktu AMR itu pertama kali keluar, tadinya TB nggak dimasukin ini, tapi waktu publikasi ya pertama keluar, TB bilang jumlah kita kan jauh lebih besar gitu. Akhirnya sekarang semua masuk gitu, dan memang kita ini nih untuk proyeknya WHO perlu lebih banyak lagi negara yang terlibat dan lebih banyak lagi tempat yang terlibat, dan whole genome sequencing will gradually. Jadi ini kalimatnya akan dan bertahap, dia replace phenotyping gitu. Dan WHO membuat platform supaya member state bisa menganalisa, menyampaikan datanya. Saya tutup dengan G20. Saya, walaupun ini bukan forumnya, tapi saya kembali ingin mengusulkan agar Indonesia memimpin penanggulangan dan diplomasi isu ini. Salah ini harusnya tuberkulosis di tingkat G20. Mau tuberkulosis stand alone boleh, mau tuberkulosis digabungkan dengan MDR boleh-boleh aja. Tapi saya sih mengusulkan kita masukkan ini di dalam agenda kesehatan, bukan saja karena Indonesia akan eliminasi, bukan saja karena 50% pasien TB di dunia itu ada di negara G20. Jadi negara G20 itu memang negara kaya, tapi 50% pasien TB di dunia ada di negara G20. Itu alasan kedua kenapa Indonesia mesti masuk. Dan alasan ketiga adalah sesudah Indonesia jadi ketua, maka ketua berikutnya India. Ketua berikutnya lagi Brasil. India dan Brasil TB-nya juga banyak, apalagi India. Jadi kalau kita nggak mimpin sekarang, kita nggak pelopor sekarang, bukan nggak mungkin nanti India yang melopor. Jadi saya menggunakan momen ini untuk kembali mengusulkan, karena kan saya bisanya cuma usul-usul aja ya, kembali mengusulkan nggak Indonesia memimpin penanggulangan diplomasi isu tuberkulosis. Dan saya sebarkan tulisan ini ke beberapa duta besar gitu ya, teman-teman yang duta besar, dan mereka sangat setuju karena dari kacamata isu internasional ini bagus sekali. Itu aja yang ingin saya sampaikan, Bu Rita, teman-teman.
Assalamualaikum warahmatullahi wabarakatuh. Terima kasih banyak, Prof. Tjandra. Ini pasti interpreternya terpontal-pontal mengikuti Prof dengan bahasa Inggris, saya tahu betul. Jadi begitu Bapak Ibu sekalian, kita masih punya waktu tidak banyak untuk questions and answers. Silahkan panitia, ada pertanyaan mana yang akan disampaikan dan kepada siapa begitu. Terima kasih Prof. Tjandra. Jadi memang setuju sekali, sayang tadi M.Kes dan Wamenkes tadi pagi, kalau hadir terus sampai siang ini, saran Prof. Tjandra bahwa Indonesia sebagai ketua G20 menyarankan untuk ini tadi diplomasi TB supaya menjadi bagian daripada global isu gitu ya. Setuju sekali, daripada nanti diambil India dan Brasil, mendingan Indonesia berprestasi dengan menyampaikan penanggulangan TB secara global. Silakan, ada pertanyaan dari panitia, dan sementara mudah-mudahan ada Bu Nadia juga di sini. Andai kata ada Bu Nadia, kita bisa tindak lanjuti. Tapi pun ada Pak Nana dari Litbang mungkin ya, dan teman-teman Litbang. Baik, ini pertanyaan buat Dr. Ristiyanto dari virus kemungkinan terjadi di seluruh wilayah Indonesia, betul, di mana ada tikus. Kondisi alam Indonesia yang sangat beragam. Ya, sekarang sedang banjir tuh di Kalimantan atau di mana ya? Bagaimana metode pengendaliannya? Misal untuk wilayah ekstrim, lahan gambut, rawa-rawa, puncak Jaya Wijaya. Silakan Pak Riz.
Ya. Terima kasih Ibu Rita, ini pertanyaan yang bagus. Dan memang betul bahwa kemungkinan leptospirosis itu bisa saja terjadi di daerah ekstrim, seperti tadi di rawa-rawa, lahan gambut maupun juga di puncak Jaya-Wijaya, terutama ini pegunungan. Yang pernah kami teliti seperti di daerah pegunungan di Kulon Progo, walaupun di sana hujannya relatif tidak membuat banjir, genangan air sedikit, tetapi memang lepto itu ditemukan pada tikus-tikus kebun, ya, seperti tikus pohon itu ternyata positif lepto, bahkan orangnya juga terkena karena dia memanjat pohon itu, bahkan sampai lumutnya pun itu positif leptospirosis. Jadi memang hal itu tempat yang ekstrem itu kemungkinan bisa saja terjadi leptospirosis. Nah, untuk pengendaliannya yang efektif dan efisien tetap aja mengarah ke manusianya sendiri. Mungkin kita nggak mungkin mengubah lahan gambut atau boleh katakan di daerah pegunungan atau di rawa-rawa. Jadi di sini untuk mengendalikan lepto itu adalah pencegahan dengan meningkatkan pengetahuan masyarakat, meningkatkan bagaimana masyarakat itu melindungi dirinya sendiri terhadap leptospirosis yang ada di lingkungannya. Dan kalau misalnya ada gejala-gejala yang mengarah ke lepto harus segera ke puskesmas seperti itu. Itu yang saat ini yang memang harus bisa dilakukan karena pengalaman-pengalaman di lapangan atau boleh katakan terjadinya KLB itu kekurang tahuan masyarakat itu sendiri maupun para petugas puskesmas atau medis untuk mendiagnosis atau mengenal penyakit leptospirosis itu sendiri. Jadi kuncinya adalah meningkatkan pengetahuan penduduk, meningkatkan kampanye atau penyuluhan PHPS tadi, dan jelas pengelolaan lingkungan agar dapat menekan terjadinya penularan leptospirosis. Saya kira Ibu.
Baik, terima kasih Pak Riz. Jadi memang lepto ini neglected atau deneglected kan, padahal sebetulnya ada di mana-mana ya, dan selalu diagnosisnya demam berdarah. Sekarang infeksi dengue, infeksi dengue itu gejala pertama, diagnosa pertama, typhoid lah. Padahal kita lupa ada diagnosa lepto. Mudah-mudahan dengan tadi Pak Riz menyampaikan adanya pedoman yang bisa dipegang oleh para dokter puskesmas, semoga bisa menyeluruh di seluruh Indonesia. Kita menemukan lepto dan itu kasusnya akan tinggi banget seperti KLB, tapi karena seluruh Indonesia paham. Terima kasih. Baik, pertanyaan kedua. Oke, untuk Prof. Tjandra dari Pak Taufikur Rahman. Prof, Perpres 851 tentang RKP, singkatan RKP, Prof tahu, Prof, tahun 2002, salah satu major projectnya adalah reformasi sistem kesehatan nasional dengan konsep penguatan health security yang melibatkan Kemenkes, Kementan, BPOM, Pemda. Di antaranya fokusnya adalah penguatan surveillance dan penyelidikan epidemiologi. Mohon pandangan akan seperti apa kira-kira implementasi dan kontribusi masing-masing lembaga tersebut mendukung national health security. Wah, tinggi nih Pak Taifu.
Sebelum yang tinggi-tinggi Bu Rita, sebelum yang tinggi-tinggi saya jawab yang gampang dulu aja. Saya mau menyapa Pak Aris karena biasanya Pak Aris berkomunikasi ke saya lewat Facebook, jadi saya senang ketemu Pak Aris sekarang begitu. Dan saya senang juga karena rambutnya panjang, entah kenapa kok rambut saya kelihatannya gundul di sini ya. Untuk
sudah mulai memutih, Prof, apa sudah mulai meninggalkan dunia hitam?
Sudah putih semua, cuman kenapa pendek sekali? Untuk pengetahuan Bu Rita, saya baru pertama kali cukur rambut sejak pandemi COVID. Beberapa hari lalu. Jadi mungkin potongannya agak terlalu kependekan. Pertama kali potong rambut sejak pandemi.
Oke.
Dan saya tahu Bu Rita suka jalan-jalan, saya baru sekali kali naik pesawat terbang sejak pandemi COVID. Pertama ke Bali dan kedua ke Pekanbaru kemarin. Ini nggak penting, nggak ada hubungan sama topik ini. Sekarang balik ke pertanyaan Pak Taufikur Rahman. Saya terus terang juga tidak tahu kepanjangan RKP apa, tapi saya tahu tentang reformasi sistem kesehatan nasional. Kenapa? Karena saya diundang waktu Bappenas membuat konsep awal reformasi sistem kesehatan nasional. Terus terang saja nih Pak Nanang, saya mengusulkan bahwa buat orang luar seperti saya tahu ada reformasi sistem kesehatan nasional, tapi saya tahu juga ada 7 transformasi sistem kesehatan yang dibuat oleh Kementerian Kesehatan. Saya mengusulkan reformasi sistem kesehatan nasional dan transformasi itu digatukan atau bagaimana begitu supaya tidak buat kita tahu yang mana yang mau kita tuju. Dan saya tahu juga bahwa reformasi sistem kesehatan nasional itu ada SK-nya atau ada dasar, mungkin tadi itu yang disebut Pak Taufik Rahman. Dan saya tahu di dalam reformasi sistem kesehatan nasional dan dalam transformasi kesehatan yang Kemenkes, dua-duanya ada tentang health security. Jadi saya kira sepakatlah bahwa health security ini merupakan hal yang sangat penting. Apalagi Presiden ngomong di sidang umum PBB, beliau bicara di KTT G20 di Milan, Italia, beliau bicara bahwa kita ini perlu penguatan kesehatan nasional, bahkan menurut Presiden kita perlu tata ulang arsitektur kesehatan global. Dan mengatakan arsitektur kesehatan global saja mesti diubah, mesti diperbaharui, mesti diperbaiki, apalagi health security di Indonesia. Jadi saya kira kalau tadi kita bilang TB itu sudah bagus, sangat bagus karena punya Perpres sebagai amunisi besar, maka health security juga sebenarnya sangat bagus karena momennya sedang bagus-bagusnya, dan Presiden bicara tentang tata arsitektur kesehatan global, tentu saja harus diikuti dengan health security di nasional. Dan yang berikutnya kita tahu kalau mau tentu semua penting, banyak faktor penting, tapi seperti Pak Taufik Rahman sebutkan bahwa surveillance itu jadi salah satu backbone yang utama. Kalau Bapak Ibu membaca, ada independent panel dari WHO yang dibuat untuk menganalisa COVID-19 ini dan apa yang perlu dilakukan ke depan dalam rangka health security. Ada 7 rekomendasi dia. Dan salah satu di antara 7 itu dia mengatakan surveillance nasional, surveillance antar negara, dan surveillance global harus ditingkatkan. Jadi sepakat sekali dengan Pak Taufik Rahman bahwa surveillance jadi backbone-nya. Soal bagaimana peran masing-masing lembaga, ya silakan Pak Nana dan teman-teman yang mengaturnya, saya sudah memberi masukan pada waktu tim informasi itu. Itu berita.
Baik, terima kasih ya. Kita mengenal ada Global Health Security Agenda, dan dulu kemarin kita membuat suatu planning semua kementerian, dan mudah-mudahan itu bukan hanya di kertas dan perencanaan saja, dan masing-masing kementerian membuat langkahnya. Silakan, ada lagi pertanyaan panitia? Oke, Prof Tjandra lagi dari Meda. Meda ini Bapak Ibu kelihatannya ya. Mohon maaf kalau salah. Seberapa besar keberhasilan program pengentasan TB melalui jalur sosial dan budaya yang rasanya lebih bisa diterima oleh masyarakat ketimbang intervensi dari sisi medis? Apa yang seharusnya diterapkan dalam lingkup sosial budaya untuk percepatan pengentasan TB? Begitu, Prof. Silahkan.
Bapak Ibu sekalian, saya sekolah tuberkulosis itu tahun 1987, saya ingat sekali. Terus 1987 itu dibicarakan tentang makanan, ini sebelum sampai sosial budaya ya, dibicarakan tentang makanan. Bagaimana peran makanan, saya ingat waktu itu ada penelitian di salah satu negara di Amerika, disangka kalau kasih makan aja orang itu TB-nya membaik, kan daya tahan tubuhnya bagus gitu ya. Kesimpulan pada waktu itu 1987 itu udah berapa puluh tahun yang lalu, itu mengatakan oke, makanan penting-penting aja, tapi kita punya obat yang menghilangkan dan yang bisa membunuh kuman tuberkulosis. Jadi obat yang bisa membunuh kuman tuberkulosis itulah yang utama. Kalau mau ditambah suplemen makanan atau tidak, silakan aja. Itu cerita waktu itu. Tetapi ini buat tentang makanan dulu saya ambil ya. Pada waktu mungkin 1 bulan 2 bulan sebelum saya pensiun dari WHO, kami di WHO berpikir ini memang makanan ini perannya besar sekali gitu ya, sehingga akhirnya dibuatlah analisa tentang tuberculosis and nutrition ini, bagaimana pentingnya peran makanan untuk mengendalikan tuberkulosis tanpa memang meninggalkan tentu saja obat. Sekarang balik lagi ke pertanyaan itu, jadi yang saya mau bilang tadinya kok kayaknya makanan tuh agak tertinggalkan begitu, cuma sekedar tambahan kalau orang-orangnya datang begitu, tapi kemudian dengan perkembangan ilmu punya peran lebih besar untuk makanan ini dalam pengendalian tuberkulosis. Begitu juga sosial budaya tentu saja ya, baik untuk tuberkulosis maupun yang lain. Apalagi kalau sedikit banyak mau dihubungkan dengan social determinant of health, itu tentu punya peran besar dalam pengendalian program kesehatan apapun juga. Jadi saya setuju sosial budaya tentu berperan Pak atau Ibu, mohon maaf, tapi kita punya tools yang kuat ya. Kalau dari tadi ngomong-ngomong COVID itu kan sampai sekarang obatnya belum ada ya. Molnupiravir yang disebut-sebut obat oral yang bisa membunuh viral aja sebenarnya kan hanya berdasarkan penelitian dari 790 sampel, separuh-separuh gitu, jumlah sampelnya masih kecil sekali. Tapi itu pun sudah digadang-gadang sebagai obat yang bisa membunuh virus. Sementara obat TB ini sudah punya pengalaman panjang, dan sesudah obat TB yang dulu disebut-sebut sebagai obat primer, sekarang sudah ada obat tipe A lagi yang beda delamanid. Jadi obatnya cukup banyak. Jadi obat itu ada, tapi sosial budaya bisa dipakai agar terimplementasi dengan baik di surveillance-nya maupun diagnosisnya maupun terapi. Jadi usul saya sih semua jalan sama-sama sekarang. Sosial budaya jalan, nutrisi jalan, tentu saja pengobatan yang Bapak bilang medis, karena bukan medis sebenarnya, ini penanganan itu jalan bersama-sama. Itu
Baik, Prof. Jadi memang obat TB ada, tapi kan yang terjadi di TB itu lama dan sakit. Apalagi MDR TB, saya dengar beberapa, saya juga jalan ke beberapa rumah sakit dengan MDR, aduh, itu yang membuat mereka bosan. Malaria yang obatnya 3 hari saja, tambah 14 hari Primapen, Pak, DO-nya tinggi sekali. Apalagi yang berapa bulan, Prof? 6 bulan, 1 tahun? Gitu ya. Silakan, ada pertanyaan yang lain? Bu, ini dia nih ahli TB, Bapak narasumber, berarti kedua-duanya nih. Menurut Bapak berdua, apakah kita sudah punya Public Health Laboratory Network? Nah, Pak Aris sama Prof nih. Kemudian selama COVID response ini, apakah menggunakan Public Health Laboratory Network atau develop network spesifik? Ini 2 pertanyaan, mau dicatat. Nanti pertanyaannya biarkan aja di layar ya, Bu. Panitia, apakah ada rencana improvement pada bantuan luar negeri ya, yang sudah ada selama COVID ini menjadi bantuan luar negeri untuk semua penyakit, untuk penanggulangan semua penyakit? Ini 3 pertanyaan. Nah, sesuai title acara ini tentang lab-based surveillance, kita semua tahu bahwa dibutuhkan Public Health Laboratory Network sebagai gerbongnya, termasuk untuk support National Health System Security. Pendapat terakhir, silahkan siapa dulu? Pak Risti dulu, monggo, nanti menjawab, setelah itu Pak Tjandra. Pak Risti tentu untuk network di leptospirosis karena tikus ada di seluruh Indonesia, dan kemudian Prof untuk TB-nya, silahkan.
Terima kasih Ibu Rita. Kalau untuk Public Health Laboratory Network itu mungkin misalnya kami pernah melakukan suatu pertemuan untuk membangun jejaring laboratorium, itu sudah kami coba ya. Tetapi kalau kita lihat dari peraturan menteri, itu memang sudah harusnya ada ini di peraturan menteri nomor 658 itu mengenai jejaring laboratorium diagnosis penyakit infeksi, new emerging dan re-emerging disease. Berarti peraturan sudah ada, jadi kemungkinan hal itu juga sudah kami rintis, dan saat ini untuk realisasinya memang perlu ditingkatkan seperti itu. Seperti kami saat ini dengan lab Karyadi, dengan veteriner misalnya, itu sudah pernah melakukan saling tukar menukar serovar, seperti itu sudah kami lakukan gitu. Jadi untuk COVID pun rasanya juga di kami ikut di dalam jejaring lab COVID yang ada di Indonesia, saya kira. Itu kira-kira informasi yang saya dapatkan. Saya kira itu Ibu Rita, terima kasih.
Baik, teman-teman. Pak Tjandra, Prof. Tjandra.
Ini sebenarnya mesti jawab. Saya gini lah, seperti Bu Rita sampaikan, kalimat yang terakhir semua orang setuju bahwa untuk pertama harus ada National Health Security System. Bukan hanya nasional, saya sudah sampaikan, bahkan ada Global Health Security System yang perlu diturunkan dalam National Health Security System. Dan untuk menjalankan National Health Security System ini mesti ada surveillance. Gitu. Tadi sudah kita bahas. Dan untuk surveillance, salah satu sendi utamanya harus lab-based surveillance. Begitu kan, itu sepakat. Tapi soal kebijakan, apakah kita sudah punya apa nggak punya, apakah yang COVID sekarang pakai public health network yang sudah ada atau bikin baru, apakah ada rencana PHLN, ini tiga-tiganya sepenuhnya adalah pertanyaan buat Bu Vivi atau Pak Nana. Kalau menurut pendapat saya, daripada saya yang jawab, saya akan keluar dengan pendapat pribadi, tapi ini kan barang yang sudah kita ketahui sebenarnya jawabannya. Jadi saya usul nanti bisa dijawab oleh Bu Vivi atau Pak Nana, bagaimana kebijakan pemerintah yang sekarang ada.
Ya, begitu Pak, Prof. Baik, sayang sekali waktu kita memang sangat terbatas, tapi setuju bahwa lab sekarang memang masih scattered gitu. Jadi Pak Bukar Melia ini menyentil labnya itu sekarang siapa yang bertanggung jawab? Sebenarnya semua yang punya lab. Ada di Dityanmed, Direktorat Laboratorium, ada di Litbang, ada di BBTKL, ada di mana lagi begitu. Ayo masing-masing, ya, kita ngiri dengan CDC Atlanta kan, masing-masing penyakit punya labnya. ya, apalagi untuk tikus ya Paris. Pasti ada, dan lab ya satu lab di Salatiga yang sangat kuat untuk vektor, ada reservoir, itu juga harus menjadi sebuah jejaring, termasuk lab di veteriner menjadi sebuah One Health Laboratory. Sayang sekali Bapak Ibu sekalian, waktu kita sebenarnya sudah lebih setengah jam tanpa terasa, dan saya sudah disentil oleh panitia. Saya akan akhiri, sebenarnya pertanyaan masih banyak, silahkan Bapak Ibu panitia yang mencatat pertanyaan dari floor. Banyak pertanyaan, sampaikan kepada para pembicara. Terima kasih Pak Ristiyanto, terima kasih Prof. Tjandra Yoga, dan mohon maaf kalau saya memang memoderatori ini tidak cukup waktunya. Saya merasakan memang tidak cukup, tapi kita tak ada yang tak terbatas. Kita terbatas dengan waktu yang mungkin harus segera diselesaikan. Terima kasih sekali lagi wabillahi taufiq wal hidayah. Untuk sesi kedua, para peserta webinar masih sekitar 300-an lebih, hampir 400 ya. Terima kasih masih stay bersama kita, dan saya kembalikan kepada MC Ibu Uli dan panitia. Sekali lagi terima kasih untuk sesi kedua ini dari saya. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Terima kasih Ibu Dr. Rita Kusriastuti atas moderasinya yang luar biasa dinamisnya, dan tanpa terasa memang sangat pendek waktunya sehingga kita tidak bisa mengakomodir semua pertanyaan yang muncul begitu. Okay, ladies and gentlemen, honored guests and audiences, I would like to remind you again about the evaluation form. Jadi Bapak Ibu, ada form evaluasi yang dapat Bapak Ibu isikan. For the in this form, informations that you fill in this form will be used for the e-certificate purposes. Jadi don't forget to fill it and give your full name and valid email address, as we will send you your e-certificate through your email address. This evaluation form can be accessed through the QR code shown in your screen, and this form will be open until 3 p.m. this afternoon. Jadi durasi pengisian untuk evaluation form ini adalah sampai pukul 3 siang ini. Setelah itu kita akan tutup linknya, jadi mohon Bapak Ibu bersegera untuk mengisi informasi Bapak Ibu di form evaluasi ini agar bisa tercetak e-certificate-nya. Okay, as we proceed both sessions in this webinar, it's nearly to close this session. I would like to give thanks highly and appreciations to our speakers and moderators, especially for their time investment and their availability to join our webinar. And for all the attendants, thank you very much for joining this webinar. If there is some inconvenience during this webinar, we would like to apologize. And would you kindly join our next webinar? Please stay tuned to the social media of Balitbangkes, and see you in the next webinar. Bye-bye.